here, Kikling? Yes. Go ahead, please.
Right. Yeah. So thank you so much, organizers, for inviting me to deliver a talk on the topic of my interest and probably interest to you. So my topic is like to develop peanut genotypes for reduced immunogenicity and heat tolerance. As you guys are aware, there is increasing number of individuals who are experiencing sensitivity to peanuts. If we talk about the United States, about 0.4% of the individuals were actually found to be sensitive in 1997. Now the number rose up to 2.9% of the population that are affected by peanut allergy. And there are 16 proteins in the peanut seeds which were recognized as the allergens. Among those 16, there are 4 proteins that are 1, 2, 3, and 6, which actually are responsible for peanut allergies, which are called like the major allergens. So about 90% of the individuals in the United States who are sensitive to the peanuts are affected by these proteins. Right now, the solution is to avoid peanuts, which is not so easy for everybody. And one has to create a peanut-free environment, which is very expensive. And the peanut allergies as a major source of allergies in the United States. I mean, the most of the hospitalization cases that happens for food allergies are generally due to peanut allergies. And the maps on the top, they were showing like the peanut allergies are very widespread. They are not confined to the areas where the peanuts are produced, but it actually affects other parts of the world, like where the peanut is consumed. So let's move on. Oops. So the two approaches that we have taken in our case, were actually, you can see there's germplasm screening and the genome editing. And the outcome of these two streams would be like the development of reduced immunogenicity peanut lines. We also wish to develop molecular markers that are for reduced immunogenicity trait. And the other thing that we would like to develop is a platform for multi-gene editing in peanuts. So we have actually screened the germplasm collections. So first screen was done with the US mini-core collection of the genotypes. And these are about 108 genotypes that would represent 10,000 genotypes that are actually existing in the US collection. We also screened the collection of about 93 peanut lines that are available in United States from the ICRI-set mini-core collection. And we have also have access to a collection of 16 lines that is South Asian collection, and they were all screened for the protein profiles. As you can see, these lines were screened through 3 different methods. One is called the polyacrylamide electrophoresis of the proteins. So you resolve the proteins on the page gels, and then you turn the page gel patterns into the densitometric analysis. We have also used enzyme-linked immunosorbent assay and also used the high-performance liquid chromatography. So all 3 methods were used to screen these materials, and we were able to identify lines that were reduced in the content of these allergens in all 3 collections. The US collection has about 25 lines that were reduced in the content of these allergens. I would like to mention none of the lines were actually lagging all 4 of these allergens. So one has to actually breed it together or pyramid the effect. The collection that is the ICRI-set collection, which is the international collection, which represent about 14,000 accessions from 92 countries, as you have seen what Dr. Singh has represented yesterday. And this collection has some overlap with the US collection, but I mean, the things are still not resolved very well. And then this South Asian collection is very unique, it contained the genotypes that were actually missing in either of these collections. So we have also access to the genotypic information for the US collection of the lines. So they were genotyped for 13,000 molecular markers. These were single nucleotide variations. And then we screened these markers, and we used protein as the trait or phenotype in this case to map the protein quantity loci. So our idea is to actually map the genes that determine the variation in the protein content in the peanut genome. So we can develop molecular markers that could be used for genetic mapping and breeding purposes. So we have done this analysis. We identified 10 QTLs that were kind of determinants of the variation in the protein content in the peanut grain. And out of these 10, interestingly, 8 of them map to the chromosomes where the structural genes for these allergens do not map. So these are trans regulators. They could be transcription factors. We decided to find what underlies these QTLs. So we looked into deeply into a QTL on chromosome 10 for ARA-H2, and we found there were several candidate genes under this QTL. We narrowed them down based on what was expressing in the seed at the right time and right developmental stage. And then we screened, specifically looked for the transcription factors that can trans-regulate the accumulation of these proteins in the seeds. And we found the signatures of these transcription factors on the promoters of the ARA-H2 genes. We, as I mentioned, we not only screened in the United States the collection of those mini core collection lines, but also the diploid lines. So we were fortunate enough to find one of the lines which is called Arachis diagoe, and there is an accession GKP10602 that was reduced in the content of these allergenic proteins. And not only reduced, I will say significantly reduced in the content of those. And we were lucky to have an introgression line population of this diagoe crossed into a tetraploid peanut. So we had access to about 121 introgression lines. We genotyped these and then phenotyped these. So phenotyping again was actually protein used as a phenotype. So we used that information for genetic mapping, and we were able to identify this is the result of the phenotyping that we have done. We clustered the RILs based on their protein phenotypes into different groups. You can see there were some high expressors of the proteins, and there were some that were reduced expressors of the proteins. And we did some detailed analysis using the mass spectrum core, so we can tell. We use that for genetic mapping. We use the KUFU approach. You have learned yesterday about the KUFU approach from Josh Clevenger. And then we use the same approach. We identified a signal on chromosome 4, as you can see, that was determining the content of RH1 and RH3 together. We looked into the candidate genes under this QTL. We again followed the same approach where we looked for the expression pattern of these genes in specific tissues and developmental stages. And then we identified the signatures of these transcription factors into the promoters of the thing. So we narrowed down to 8 candidate genes that we are now in the process of validation. We identified the reduced immunogenicity lines or introgression lines that we decided to actually cross together to pyramid their protein phenotype into the thing. So we made several crosses. I'm giving you examples of the 4 of the populations that we have created. And you can see we were able to identify lines that were actually pyramided the effect. So out of the 4 crosses, we obtained about 106 F2 lines. They are kind of like the conventionally bred materials where we use the protein as markers and pyramided the protein phenotype. My favorites are in cross 1 and cross 4. You can see there were lines that were significantly reduced in the content of ARA proteins, as you can see right here. I don't know if my cursor is visible. No, probably not. Okay, so we had access to another population, and this genotype, which is called NC25, also stem out of our initial analysis of the reduced immunogenicity lines. This population is a cross between NC25 and Georgia green. Georgia green was also identified to be reduced in the content of RIS3 and iso-RIS3. And we had access to this population through our collaboration North Carolina State University. We had access to about 176 recombinant inbred lines, which are already at the F6 and F7 generation. We screened them for their proteins, and you can see they were segregating for RH1 free. My example doesn't show the segregation in RH1, but they also, believe me, they also segregate for RH1, RIS3, iso-RIS3 content. We use that for genetic mapping. This is the heat map showing the distribution of the proteins in different recombinant inbred lines. We use that for genetic mapping. We found signals on different proteins, different chromosomes. They were both cis and trans regulators of the gene. So some were mapping close to the genes, the structural genes responsible for RH1, 2, 3, and 6, and some mapped away from this. We also performed the Western blotting of these lines, which we liked. And this Western blotting was done through the antibodies that were developed specifically for RH1, 2, 3, and 6. This was done in collaboration with University of North Carolina, where the people who have developed the oral immunotherapy has helped us do this analysis. We also use the human serum immunoglobulin E's. These are derived from the peanut sensitive individuals. And these immunoglobulin E's specifically bind to these proteins, as you can see in the line 317 shown on the far, say like left of you, but actually showing reduced cross reactivity. So this line seems to be very promising, and it will be analyzed using the BET assay, like which is a gold standard to find if these cause sensitivity in individuals. We also use multi-gene editing approach. As you can see, these genes exist on the 6 different chromosomes to 3 homologous groups. We pulled out their sequences from the database. We aligned them and identified the conserved sites in these genes. We developed the guide RNAs, which guide the Cas12a enzyme to the right site. We tested that in vitro using the gel-based assays. You can see it will process the product into small products. We also use a chip-based assay, so you can use a chip like that tells the difference in the electrical conductivity when the DNA is processed. We developed a multi-gene editing construct using this. So we developed 3 different constructs at the moment. Could you help, I won't remove this. I'm sorry for this interruption. Oh, thank you. I'm very sorry for the interruption. So we had the 3 different constructs that were developed. So the first construct that you can see, PAKB005, is actually having hygromycin resistance. So this is an herbicide, like antibiotic. And then 006 has GFP. And then we had developed recently a third construct. We first transformed this into the protoplasts. So these protoplasts are the cell wall-less cells of the plant, and they're easy to transform. They are very much like the animal cells, which are not having cell walls. So we use polyethylene glycol to transform this with the construct that contain GFP, and we determined about 56% of the transformation efficiency that was determined based on the 10 different experiments that we have performed. Then afterwards, we actually tested it for the mutations at the target site. Remember, I used the Cas12a, which can process the multi-RNA transcripts. So we found edits at all of the sites. It being Cas12a, it produced deletions. So we identified the deletions, which were actually pretty large. As you can see, they were actually about deletions of 11 to 17 nucleotides at the target site. We confirmed those results using the Sanger sequencing method, because the next generation sequencing, you use the pools of the DNAs. We deconvoluted using the Sanger method, and we found that the messed up peaks that you were seeing is a method developed in the Daniel Voitas's lab in the United States, where they found if you see these messed up peaks, it's because of the edits at the site. After testing these reagents in the protoplast, we delivered using a special method that we call the vacuum infiltration of the viral construct. This is kind of a strategy derived from the human editing experiments. So we actually use a tomato leaf curly virus, which can carry a very large cargo of about 6 kb. So it can carry the Cas12a as well as the guide RNA. So we delivered it using the vacuum infiltration of the intact peanut plants, and then the gels were showing the delivery of the construct into the plant. You can see the fresh tissue was showing the GFP amplification. Okay. Now the panel was showing the delivery of the GFP in the xylem vessels of the plant. As you can see, the xylem vessels lit up with the green light, and that's because of the GFP. We confirmed the GFP by DNA sequencing. We analyzed the target site for mutations in these ARA-H1, 2, 3, and 6 genes. We had the similar results. We found the edits of about 1 to 6 nucleotide deletions. We also used this by the biolistics approach, which is the gold standard of transformation, but it is dependent on the tissue culture. So we did tissue culture. You can see it takes awfully long in case of peanuts, like it's about a year from seed to seed. So we transformed this material. We did some early analysis by sectioning the calli with the GFP. We confirmed the GFP delivery using the bialistic method into peanuts. Now I'm showing you the results of the multi-gene editing in the peanuts. And you can see like it actually showed a location effect on the processing. Like, so we get more edits in RIH1 and RIH6 in comparison to RIH2 and RIH3. So it kind of tells like there is a processing difference between the transcript. So because as I said, like it's a multi-gene editing construct. So it has like kind of guides for different Arakin genes. We obtained the T0 plants, obtained the T1 seeds. We studied them for their protein profiles. So we split each seed into two parts, the embryo half and the cotyledonary half. The embryo half was kept to produce a plant. The cotyledonary half was produced for the protein analysis. We profiled their proteins. We were showing like the heat map as well as like the constellation plot. But you can see there were lines that I really liked, especially the 29 series of lines which were actually lacking specific proteins from the seed. And these were the allergenic proteins for RIH3, iso-RIH3, and RIH2 and 6. We did analysis, alike analysis for the T2 generation, that's the next generation. And we did the same strategy. We split the seed, we analyzed for proteins, and the 29 line that was my favorite is still carried the phenotype. So it's transmitting the phenotype to the next generation. Now I will switch gears and talk about the heat tolerance. So we did profiling. We did profiling of the anther lipidome. So we wish to use the lipids as the biomarkers. So we decide to study the lipid profiles. The lipids constitute the membranes in the plant. So and they help the plant to acclimatize under the heat and cold. So we decided to analyze it for the lipidome. So we did is study the 82 species of the lipids in the membranes, and we noticed like the unsaturation index of the lipids reduce under exposure to the heat. And that's probably the acclimatization mechanism. I'm sorry. So we studied the expression pattern of the FED3 genes, which are responsible for turning the diacylglycerol chains into triacylglycerol chains. And then we saw like a reduction in the content of the FED3 as well. We had access to a RIL population from collaboration in Texas A&M. It's a small population, but we screened it for the thing, and we noticed this is a cross between a resistant line, which is ICS 76, again the origin from ICRISET, and then it's crossed with a Texas line, which is the high oleic acid line, and we screened it. We noticed like the same sort of reduction in the case of the plastidic lipids and extraplastidic lipids. So the content of the lines which are highly lipids which are highly unsaturated reduced with the heat stress, and that could be used as a marker like to find the changes. We also found an interesting pattern that triacylglycerol and estrol esters increased. You might wonder why, like the membranes remove these highly unsaturated lipids and they are sequestered in the form of these two species of lipids. And that reduced the oxidative stress on the plant. We classified the RILs based on these lipid signatures, and then we found the lines that were heat tolerant mapped with like or clustered with the ICGS 76, and the ones that were kind of susceptible clustered with the other parent. We use this for genetic mapping, again, use the KUFU approach. We identified the QTL chromosome 16 for this that tells like this is a regulatory gene which can be induced with the heat. We use another molecular, another kind of approach where we use the pollen viability. We use to screen like the 72 lines of the mini core collection. We compared them with Georgia green. This is a popular variety from Georgia. And then we compared the worst and best performers, and we used this data for genetic mapping. We found a QTL on chromosome 20, which aligned with the QTLs earlier identified by the ICRISET crew. So with that, I would like to thank my group, my collaborators, and will be happy to address any questions you have.
you. Thank you, Sachin. We can have one question if the audience is. Yes, please.
Yeah, it's impressive work. So my question is related with your gene editing. What kind of transformation you are apply for your research? Because we know the peanut transformation very difficult.
Yes. So the question is regarding like what transformation method we use. We use two different methods. So we use like one is the viral based delivery use of the intact plants via vacuum infiltration. The other method we use is the particle bombardment or biolistics approach, but it's dependent on tissue culture. So we use Georgia green as a source of the calli, which was demonstrated in Peggy Ozias's lab. So it's an established procedure that we have adapted from her. It worked well, but it takes time.
We have to stop here because we have to take care of the time. Our next speaker is Dr. Kyung Liu. He is from CRI, China. We're talking to us about progress on peanut genomic research and breeding applications at CRI. This is Guangdong Academy of Agriculture Sciences. So we have this recorded message is at the online. Recorded.
ये तब
这个是能新手上吗？是吧？是特殊的。这个是塞内加尔一个。不是吧？隔音电话，这个是。昨天他不是还那个？哦，这个是。
Today, my topic. Hello, ladies and gentlemen. Firstly, thanks to AAGB meeting group to give me this opportunity to make my presentation. I'm Lu Qing from Crop Research Institute, Guangdong Academy of Agriculture Sciences. Okay, let's start my presentation. Today, my topic is about progress on peanut genomics research and breeding application at CRI-GDAS. And I will introduce my presentation from the four contents. has an impact on peanut is one of the important oil crops in China. So oil biosynthesis is the focus of our attention. We perform a GWAS analysis for oil content traits and identified a candidate interval on chromosome A8. This region was focused on a 74 kb. Through LD block analysis, we found only one gene was highly homologous to linked one gene in apoptosis. This candidate gene is very clearly involved in lipid biosynthesis in apoptosis and other crops, such as soybean, maize, and rapeseed. We consider it was a candidate gene and named it as HWI1. This candidate gene was highly expressed in seeds compared to other tissues. And overexpressing in apoptosis showed that it can highly increase oil content in seeds by regulating oil body size. But how to apply this reference genome or population genome findings? We constructed a consensus map using the reference genome of 2 wild species in 28 and detected 40 meta-QTLs. We also developed 970,000 new SSR markers in cultivated peanut genomes in 2019. These SSR markers would be helpful for mass or cultivar mapping. Moreover, we developed a liquid chip containing 40,919 SNP markers. This chip would be benefit for cultivar mapping, background selection in breeding or GS breeding. Finally, we also conducted gene editing for oil content gene HWI1. And summary, we released the genome of cultivated peanut and its 2 wild diplo progenitors. We sequenced 319 associations worldwide and constructed a genome-wide variation map. We performed a GWAS analysis for 28 agronomic traits and identified thousands of significant associations and identified 2 candidate genes. We also developed a total of 973,984 SSR markers that can be assisted in gene mapping or mass breeding. We also developed a liquid chip that can be used for cultivar mapping, genomic selection, or mass application. Finally, a gene editing is currently underway. All of these results were published on Molecular Plants, Peanuts, and Nature Genetics. These above achievements were supported by different fundings, such as NSFC. Finally, we also sincerely thank to all collaborators for their help. Thanks for our group members. And also thanks to all. Thank you very much. That's all.
Thank you. Thank you very much, King. It was wonderful listening to you. Would have been very happy if you were here in person. Have interacted more with you, but we probably missed that one. So we next go. I'm told the next speaker will be Bashao Liao, and he'll speak to us on identification and utilization of genetic variation in bacterial wilt resistance in groundnut. So, is he online or is it a recorded one?
China. I'm Bu Xiong from the Oil Crops Research Institute of the Chinese Academy of Agriculture Sciences. Firstly, I
Lot of till we see many challenges. Oh yes, we have Basha here now.
yes, I'm here. Morning. I just wonder if it's not apply. I can also apply the record one from my country here. This 4 parts including introduction, updates of resistant discovery, improvement of resistance, and the Chinese Academy of Agriculture. My presentation today. However, the groundnut production in certain regions is still affected by bacterial weight, a soil-borne bacterial disease. This important disease has been a problem in Southeast Asia and Central Africa, including China, Indonesia, Vietnam, Thailand, and even Uganda. Even the Rastonia species affects many crops in India. India is largely free from this disease. As you can see, the picture. As you can see, in China. Sorry, I think you're planning the two files together. There are 2 copies, there are 2 visions. My presentation today contains these 4 parts, including introduction, updates, resistant discovery, improvement of resistance, and a summary from the Academy of Agriculture Sciences. Firstly, I would like to express my thanks to the organizing committee for giving me this chance to deliver a presentation at this important conference. My presentation.
Hi, Basha, can you come online? We would like to listen to you in time. Basha, you can hear us?
Yes. Contains
No,
these four
probably.
parts, including I don't know what's right then. Or if it's I can still, I can play here.
Doesn't matter, not to be panicked. In case there's an issue, we can contact him. Oh yeah, he's here. That's good. So Basha, you can make your presentation from your side.
Yes, I can play it. I can play from my
We are not
computer.
able to listen him. What's about the sound? Can you? There's a cross there. Is that sound? Yeah, nothing to worry. It's we would like to listen to you and let us.
Okay, I can just play from my computer here. Try it.
Yes, that's fine.
Hey,
So
yeah,
who's going
so
to make the
sorry.
presentation from here or from his side?
Yes. Yeah.
So you can load presentation from your side.
Yes.
Yeah, please go ahead.
Yes. It's very strange. So I can play here. Can you hear it? Ladies and gentlemen, this is a greetings from China. Can you hear it? I'm Hu Liao from the Oil Crops Research Institute of Chinese Academy of Agricultural Sciences. Firstly, I would like to express my thanks to the organizing committee for giving me this chance to deliver a presentation at this important conference. My presentation today is identification and utilization of genetic variations in bacterial weight resistance in groundnuts. My presentation today contains these four parts, including introduction, updates of resistance discovery, improvement of resistance, and a summary. As you know, groundnut is widely grown in the world. During the past decade, the harvest area, yield, and the production all increased dramatically, among which the global groundnut production increased from 46.28 million tons in 2014 to 54.27 million tons in 2023, increased by 17.26%. During the same period, the groundnut production in China increased from 16.48 million tons to 19.23 million tons. And in 2023, the Chinese production accounted for about 36% of the global production. However, the groundnut production in certain regions is still affected by bacterial weight, a soil-borne bacterial disease. This important disease has been a problem in Southeast Asia and Central Africa, including China, Indonesia, Vietnam, Thailand, and even Uganda. Even the Rastonia species affects many crops in India.
Dr. Bashu,
Yes.
just a reminder.
Groundnut crop.
Yes, Dr.
Yes.
Bashu, your screen is not visible.
Weight can cause serious yield loss in the infected
看不
fields.
到 PPT，只能看到这个桌面。
看不到 PPT
对，
对你刚才可能鼠标移过去了，还是
啊？
你看你到这边来。
Yes. Thank you so
Okay.
much.
Sorry.
Please proceed.
In China, about 15 to 18% It's of all right. the groundnut area is infested with a bacterial weight pathogen. Up to now, the bacterial weight in groundnut could be only effectively controlled by planting resistant covers. And breeding for this resistance has made much progress in the past 40 years. However, three challenges still exist in the genetic enhancement for bacterial weight resistance in groundnut. The first challenge is the low yield potential of the available resistant covers. even the yield level of the improved varieties increased somehow compared to that of 40 years before. The yield potential is still relatively low compared to the susceptible countries. The second challenge is the undesirable resistance level and the stability. The third challenge is lack of enough resistance to other soil-borne diseases such as stem rot, powder rot, root rot, and even asparaginous infection in the available bacterial weight-resistant coverage. Under the above background, the team at the Okricas has continued our research efforts on improving the resistance by conducting different projects, including projects cooperated with EcoResearch. The second part of my presentation is updates of bacterial weight resistance discovery. Totally, over 150 genotypes have been identified as resistant to bacterial weight. Among the 6 varieties of recultivated granite, resistant germplasm accessions have been identified from 5 variety types. Mostly resistant lines belong to the Spanish type and the Isuta type. In addition to this, 300 wide arches accessions were tested for their relation to bacterial weight, and 16 accessions were highly resistant. Some accessions of the right species with a high level bacterial resistance may possess stronger mechanisms in resisting the infection of pathogenic bacteria from the soil. Based on microbiome sequencing, more diverse bacterium were found in healthy groundnut plants compared to the diseased plants, and the identified bacteria might inhibit the increase of the pathogen inoculum. The reproduction of the pathogen root is much faster in the susceptible genotypes than in the resistant ones, and thus the key mechanism of the resistance is inhibiting increase of the pathogen inoculum after infection. The inhibitor effect on pathogen inoculum increase in the resistant genotypes might be achieved by enhancing expression of metabolites such as alkaloids. Zhonghuan number 6 is a resistant carver with bacterial weight resistance transferred from an landrace belonging to Spanish type. It is also resistant to powder rot and even aflatoxin production. Through trials by grafting root and stem between resistant and susceptible genotypes, the results indicated that the root of Zhonghuan number 6 is a clear tissue resistance function. ICG 12625 is a landrace from Central America. It is resistant to bacterial weight, aflatoxin production, scable, and foliar diseases. Interestingly, this genotype was found to process only 1 gene for the symbiotic alternation, and its bacterial weight resistance might be correlated with the relatively poor root system and lower yield potential. HP17 is an introduced landrace belonging to Valencia type with resistance to bacterial weight, stem rot, scable, and foliar diseases. 9102 with bacterial weight resistance transferred from Arachis diagonally. Is also moderately resistant to pollutant. When we compare the two subspecies of the cultivated groundnut, the resistance level against the bacterial weight is stability, time of disease symptoms, root system, and even nodulation case exist wide difference. This might imply that there is great potential in recombining the different resistant genes of nocell for improving the resistance, stability, and even yield potential. The third part is about resistance improvements. Up to date, we have identified major cultures regulating the bacterial weight resistance in 4 resistant genotypes through linkage analysis. For more accurate and quick phenotyping for identifying cultures, a novel resistance screening technology by hydroponic culture of the groundnut plants and the inoculation of the plants by aiding pathologic solution was developed and used. Among the 4 major cultures identified from 4 different resistant genotypes, including Zhongguan number 6, Yuanzhang 9102, Fuchuan Dali, and the ICG 12625. The one from Zhongguan number 6 on the chromosome B2 has the highest phenotypic value expressed, briefly PVE, and the genetic effect. The PVE of the culture from Zhongguan number 6 is as high as 50%, much higher than those from other resistant genotypes. Moreover, through a GOA analysis of a nature population consisting of 400 diversified adaptations, a candidate R gene on the chromosome was also identified, and its SMP variations changed the structure of LRR domain. The above markers could be used in breeding selection, even as diagnostic markers for resistance. In addition to bacterial weight resistance, cultures for other traits including aflatoxin resistance, oil content, oleic acid content have also been identified for breeding use. By applying identified markers in fake cross breeding program, any successful cultivar could be invited into resistant one within 5 years. Several new resistant cultivars have been developed and released for production. Zhonghua 212 is a bacterial weight resistant cultivar developed from a cross combination consisting of parents belonging to both Spanish and Virginia types. Its yield potential, root system, yield tolerance, and even direction to stem root are much better than most other bacterial weight resistant cultivars. Another progress is by using mass-based fake crossing breeding strategies. Zhonghua 21 was successfully invited into Zhonghua 29 with desirable bacterial weight resistance and high oleic acid. This new cultivar is popularly used in central China. Extensive application of improved bacterial weight resistant cultivars has greatly contributed to the integrated management of this soil-borne disease. and hence contributed to the increased productivity of groundnuts in the vast regions naturally infested with Ralstonia solanus rare. In summary, bacterial weight remains a key constraint to groundnut production in central regions, and better resistant categories are needed. Diversified gene plasmatizations with bacterial weight resistance have been identified both in the cultivated species and wild species. Different mechanisms for resistance to bacterial weight might exist among the diversified gene plasmid connections. Genomic polls have promoted the breeding for bacterial weight resistance and highly important traits like aflatoxin resistance and high oleic acid. In the future, further research efforts for recombining resistant genes of cultures from different subspecies are highly crucial for improving the bacterial resistant level and its stability. Special emphasis should be given to discovering resistant genes existing in various wild Arctic species. More research should also be undertaken for improving the root system and the symbiotic nitrogen fixation capacity for the bacterial weight resistant coverage for enhancing the yield potentials. Molecular breeding tools needed to be further improved for integrating resistance to bacterial weight and to other soil-borne diseases. Finally, I would like to give my acknowledgements to the team members at Okra Cast and the collaborators from EcoSet for non-PhD support of research for enhancement of resistance to bacterial weight in growth. Thank you very much for your attention. Hope all of you have a nice stay in Goa for a conference. Thank you.
Good.
Okay, that's it.
Thank you. Thank you. Thank you very much, Basho. We can have one question from the audience. Anyone wants to? Yes, Basho is online.
Yes, I'd like to.
Anybody from the audience? So if not, Basho, I would like to have one clarification. You said there's a possible relationship between the bacterial wilt resistant lines and the nodulation. Is it a
Yes.
is a common mechanism? Is very well established or still a speculative one?
For now, history from all observations, the situations like this, the bacterial weight resistant gene plasma for developed varieties, if they are resistant, Spanish type, the root, the top root is short, it's narrow, and on the major roots, there were very few nodules. Because the rhizobia is also kind of bacterial. So we guess there is a common mechanism for the resistant genotype to resist the infection of a bacterial with pathogen as well as the rhizobia strains. That is why in most of the bacterial-resistant ground varieties, the yield potential is much less compared to the susceptible ones. So in the future, we will try to make sure what is, maybe there is a common mechanism.
Good. Thank you. Thank you very much. I would have loved to have you here in person, but nevertheless, it was very impressive work, very impressive teamwork. Thank you once again. Thank you very much.
Thank you.
So
Thank you.
after two online presentations, we have now Rayan here in person. Ren, the floor is yours. You can really make a change now here. And Ren will speak on characterization of a major QTL that's influencing the shell strength in Virginia type of the peanuts.
Yeah, thank you very much. And I would like to thank the organizer to give me this chance to speak with you today about a trait which is a shell strength, the genetic of shell strength. This work is done mainly by my great student, Gilad Ben Israel. And why we are working on shell strength, and the reason is that the peanut industry in Israel is not a commodity. The industry is very small and it's pointed for within shell markets to Europe. And in this kind of market, we are concentrated, this is like 70% of the growing peanuts in Israel. There is another 30%, which is for shelling, but mostly it's for within shell to Europe. And in this market, we want bright, golden, and smooth Virginia type pods, large and super giant, and also of product is actually a trademark in many European markets, many in Italy. So, as you can understand, we actually sell the European shells. So the main problem is breakage of the pods. And when it happens, so less pods can be marketed to Europe, which lower the revenues for the growers. So shell strength is the main characteristic that affect the shell breakage. Also, pod hardness is also important to mitigate other diseases. So shell strength has genetic background because varieties are, there is a difference between different varieties for shell strength. However, the genetics of this trait is never almost never studied before. And mainly this is usually we used to, it's by manually. A person just break the shell and decide if it's hard, soft, or medium. So the goals of our research, Gilad's research, was to develop a quantitative and objective method to measure shell strength and to study it in the real population that we have and to map it and try to find significant QTLs. So the first goal was to develop a system for measuring, quantify shell strength. And we used the texture analyzer for this. We tried many probes and many different ways, complete pods, isolated shells, and eventually we found that the best way to measure shell strength is by using the P5 probe and isolated shells. And when you do that, so the breakage point, the newtons that you need for the breakage point, this is our measurement value. So as you can see here, this is the two parental lines of our population. Hanoch, which is within shell peanut market, which is a very strong shell, and Harari, which is used in Israel for a shell, local shell market. As you can see here, the nice difference and also the R value. So we have now a good way to measure shell strength. So next, we just went to existing real population that we have between the two parental lines. They were checked in two different environments, in blocks design, and 25 mature, really mature and dry pods were taken from each line. And we used existing genetic map that it's based on the peanut chip microarray that we already used it before for other traits. So this is the phenotyping to environment. You can see here not so much difference between environments, and actually ANOVA tests show no difference between years. And also the heritability rates were quite high, which means this trait has a strong genetic background, and therefore we just combine all data from the two different environments and ran one QTL analysis. 3 QTLs were resolved. One of them was with quite high significant value. We can say it's a major QTL. It's almost 20, it can explain mostly 20% of the total variation. This was on BO2 chromosome, on BO3. There's another one. These two were derived from the ANOH parent, which is the hard shell parent. The other one, the third one, is actually derived from the parent with the soft shell, the other parent. So next, we had now a major QTL, and we would like, and this is a nice story, because we would like to know where it's originated from. And what you see here, this is like the last 50 years. This diagram, it's for the last 50 years of peanut breeding in Israel. And you can see that there are two main origins for what we say within shell peanut market, not the shell market, but the soft. One is this one, this 113, which is the parent of Harari. And another one, it's in the green, is the Florispan, which is actually Spanish. So it could be one of these two, because Spanish type usually has softer and lighter shells. So first we thought that maybe the Florispan, this is the origin for this soft shell. However, we tried to look all over the peanut collections. Is, for example, one collection that was recently published by Jen et al. In Nature Genetics. Most of you know this work. You can see here all 355 genotypes. And when we are looking on all these fastigiata types, so other traits in our population, like branching habit, flowering pattern, and early maturity, came from this origin. However, our major QTL is not from there. And actually, it's not this word collection of 355, and none of them has this, the allele, the Harari allele, yeah, the allele for soft cell, even none of the Virginia types, so where it came from. And the answer came from our history of the lines. So line 113, the one that later will generate Harari, it came in the early 80s from North Carolina. This is the Isaac Wallerstein. He was the breeder back then. So we went to another study made by Jeff Dunn lab in North Carolina. As you can see here, they also sequenced Bailey 2 and made resequenced the whole history of breeding, the breeding history in North Carolina. As you can see here, our main QTL, the allele of Harari is in green, and the allele of Hanoch is in yellow. And you can see here that part of the history of North Carolina, including Bailey, here, Bailey on the side. On the side here, on the green, you can see Bailey and Bailey 2, and all the NC9, NC7, going back to Jenkins, Jumbo, variety, all of them has the Harari soft shell. So now we think that the soft shell is a negative trait that arrived into the Israeli germplasm from this origin. So we did other experiments now I'm going back to the QT analysis, and out of this analysis, we took two extreme phenotypes and genotypes. One, it's very hard, as you can see here in this picture, AAGB. It's very hard. It's a real line which it has very hard shell, and another one with a soft shell. And we analyzed them further, as you can see here on the upper left, shell strength, the difference between the parental lines and these two reels. And actually, the weight of this shell was higher, as you can see here on the right. However, the thickness between the parental lines there was no difference in the thickness, as you can see below. And but H115 is also more thicker shell. Next, we were looking for some microscope dissection. As you can see here on your left, this is the hard shell, and this is standing for lignin. And the hard shell has higher lignin content than the softer shell on the right. So now, with little time that I have left, I will show you how stronger shell actually can help you to mitigate other problems. Like here is the disease, the white mold or stem rods that all of you already know about. And this is an experiment that was made for 8 genotypes for this disease with inoculation. So the first phenotype in this, it was with the blocks and reps in field conditions. So the first phenotype is actually how much the plant is dead. We have from 1 to 5. These were 8 genotypes that are ranking also for white mold resistance and also for shell strength. So when you're looking just of if the plant is dead or not, or the disease level, you can see here that no any correlation to shell strength. And you see that HH115, the one that with extra strong shells, is somewhere in the middle and no any correlation. But another way to check the phenotype of resistance is to look on the actual pods or yield in this case. This yield is for infected or non-infected in blue and red. And when you check the actual yield, then you can see that HH15 has a higher yield, so it's less infected directly on the pods. And this is good, also for what we say weighted yield and infected yield. So future work right now, Gilad is trying. So I'm going back to the main QTL. So we were not so lucky in the beginning because the microarray in this location, this region, was not have many markers. So the insert right now is 15 million base pairs. However, we were lucky because this segment actually came from Bailey 2, which is sequenced, of course, and the Hanoch is similar to the TIFF runner. So we now, although these were not on the chip, we found many SNPs. Out of this, the most cost-effective for now, right now, is making 48 cast markers already designed, and now Gilad is going to re-genotype the entire population with these markers. Also, Gilad is now building neuroisogenic lines based on this segment. It started actually with the real line that has already 77% of the RRA genome. We decided to do the RRA genome, and the segment came from the Hanoch. because to be positive, you know, that we are increasing the shell strength and not decreasing it. Also, we are looking for candidate genes, and one of the genes that came up, it's what we call NAC1 gene that show in pumpkin, showed there is a mutation for cellular biosynthesis called the Hulles seeds. And we saw this is currently one of our major candidate genes. And also, this is the last slide. Gilad is also now looking for other populations. So 2 populations also existing with existing map in our lab. One of them is the new one on the left, is with Congo. Congo is a Valencia type, is fastigiata. You can see here it has lower shell strength. And so this is a new source to checking this trait. So with that, I will finish. I will thank you. This work was mainly done by Gilad and also William Lelwa. It's another PhD student. He is the white mold person in my lab. So with that, I will finish, and thank you also the other collaboration and you for listening.
Thank you. Thank you very much, Ryan. We have time for 1 or 2 quick questions. Please stand.
Okay. So for who? For the farmers, perspective of our farmers, harder is better. They want as much. No, for the customers, you know, our peanuts right now are too hard anyway. However, most of the people, they break not like we are expert. We are opening like this, but most of the public open the peanut in the middle. And because of the long shape of our product, it's not so bad. So if it'll be a little bit harder, it's not a big problem. Yeah, I cannot hear you.
Hi, Ron. It is a well-designed experiment. I really enjoy your talk. My question is related with the G by E interaction based on your ANOVA table. You have a genotyping by year, but I was wonder if you are doing in a different calcium content field, what will be?
If I did what?
Like a G by E interaction for the ANOVA table, that they doesn't showing
I don't remember. I don't think we show it. In this case, we had 2 environments, and the data was very, very stable between the 2. So we didn't have any environmental interaction in this case.
But I was wonder if you put it on a different soil type.
Different soil, yeah, I didn't say. Totally different soil type. One in the north of Israel, which is heavy soil. I didn't have so much time to say all this. And in the other one is in sandy soil, totally different soil. We think in this population, this trait is very stable genetically.
Okay.
Almost like seed size.
Thank you.
Dr. Ram, can I
Excellent
ask you a
presentation,
question?
Ran. We enjoyed it. I have one question regarding the cell strength and then aflatoxin contamination. Is there any correlation whether the stronger the shell then will give a resistance to the aspergillus flavus? Though the pod will be infected, but if shell is hard, then the toxin will not be produced in the seed.
So I know from other people it's interesting, and they think there is. Because we are in Israel irrigating, this is one of our big advantages, that we almost don't have any cases of aflatoxin in peanut. This is what makes us, this is also another advantage of ours. So we never check this.
Yeah, also any genotype with the stronger shell, I mean this with the good shell strength, you can check for the infection percentage. Yeah, thank you.
Rehan, congratulations for one of the rarest kind of work. I'm curious to know the higher or the tougher shell has got any lower shedding percentage, number one. And
What? Low?
yeah, like low shelling percentage. I
The
mean, low shelling
percentage?
percentage. Yeah. Tougher the shells. Did you observe any correlation in
You mean seed percentage or shell percentage?
shelling percentage?
Yes, shelling percentage. It has lower because the shell is heavier, but we don't have any problems with pod filling. Pod filling is okay, but the shell is thicker, so the ratio between shell and seed is lower. Seed and shell.
Yeah, thanks. And second question is like how the value chain operates in Israel. Whether do you have any demarcation, say for in-shell markets separately and then shell
Yeah, we
markets
have it's two
separately?
different markets almost. Like 70-80% is within shell. This is being growing in the south of Israel on sandy soil, and the pods are bright because it's on the sensor. So this is for marketing. On the north of Israel, it's heavy soils, and it's more for shelling. So it's almost totally different market.
Sir, I have a similar question what sir asked about shelling percentage. What is more desirable? A thicker shell, thicker and hollow shell is more desirable, or a thinner or a heavy shell is more desirable in Israel? In India, shelling percentage has an important value while breeding. So I
Yeah.
want to your opinion on this. Thicker shell but hollow, thin shell but heavy. Which one is more desirable for
We
shell
want
strength?
both. We want thicker shell, and the filling and good filling is very important also for us. Every market for within shell, because if the shelling percentage is not good, the pods will get burned in roasting. So it's both of them are very important. I don't see any difference between them. Can be both.
Well, thank you. Thank you, Ron. Those who have some more questions, we can have one-to-one interactions. We cannot continue beyond that. Our next speaker is Virendra Kumar. Is Virendra around? Yes. Good, Virendra. And Virendra will be talking to us about the stem rot resistance and the novel sources of resistance for that one. Floor is yours, Virendra.
Okay, good morning, everyone. Now I'll be presenting about stem rot disease, which was a minor disease before. Now it is becoming major, and it is seen in all the growing parts, all the groundnut growing regions. Stem rot disease is caused by Sclerotium ralphsi, which is a soil-borne pathogen, which is a facultative parasite that kills the plant first and then gets its food from it. So it is known as facultative parasite. And this produces sclerotia that stay in soil for almost 25 years. That means it will be waiting for the favorable condition. Whenever it gets the favorable condition, it just grows and then it infects the plants. So crop rotation will not be answer for this, so that will not be a good solution for this. And as it is a soil-borne pathogen, it is very difficult to manage, and a drenching of too much chemical will degrade the soil properties and soil health. So this must be managed, and one of the best solution is genetic management, that is host plant resistance. So the symptoms of this disease are like initial yellowing, and then the white fluffy mass of mycelium will be grown on the ground and stem parts, and then the brown lesion appears, that is the death of the cell, and after that the wilting occurs. Due to the death of the cells, there will be no transport of nutrients and water to the plant, so that the wilting occurs. And in the photos, you can see the symptoms. The first photo indicates the mycelium growth in the Petri plate, and after inoculation, the second photo indicates the growth of the white mass of mycelium that is grown on the stem. And then in the third and fourth picture, we can see the symptoms, how the devastating this disease is. That means it will lead to death of whole plant. That means it will lead to yield loss up to like 80 to 100%, even 100% has been reported in the literature. And in the middle of the last two photos, we can see the sclerotial bodies that have been formed on the stem that will be at the collar region. And these are very dangerous because they can stay for a long time. So, and after that, when it comes to mechanism, how this infection occurs through sclerotium, like how this sclerotium ralsii infect the groundnut plant. We can see when the healthy plant is there, the can I think this is fine. This is fine, yeah. This is the healthy plant, and this is the stem, how it looks like. That means it is green and it is strong. And this is how the inside view of the cell, the tissues look like. And this center region is the vascular bundle. When the sclerotium ralsii infects the plant, it produces lots and lots of mycelial mat, and the mycelial mat will secrete the oxalic acid. That is the primary agent that is produced by the sclerotium ralsii to create the death of cells so that it can feed on it. So it produces oxalic acid which disintegrates the cell. After disintegration, the oxalic acid will enter the vascular bundle and it forms calcium oxalate crystals. These calcium oxalate crystals will block the vascular bundle so that there will be no transport of water and nutrients to the bodies, like plant. After that, the ROS occurs, then the death of whole plant occurs, like all the tissues occurs. After that, the stem looks like this. That is, it is discolored due to death of cells and the stem is weak. After that, the stem falls off. This is the mechanism of sclerotium ralsii causing stem rot disease. So, to know whether we have resistant sources for this or not, that means we can use post-plant resistance in this particular disease or not. We went for screening, and this was the screening procedure followed. That was mass multiplication of pathogen was done, and it was inoculated in the field, and then the disease scoring was done based on that. And the material used for this particular trial was 184 minicore genotypes were used with the check of CS19 as resistant and TMV2 as susceptible. And this CS19 is a moderately resistant check, and TMV2 is a highly susceptible check, which is having like more than 90% of mortality. So the design use was alpha lattice, and inoculation was done at different stages, that is at 20 days of sowing and 45 days after sowing. The observations were done for two component rates, that is percent disease incidence and percent mortality. That is number of death of the plant is percent mortality, and incidence is how many plants are infected. Then the scoring was done at 30, 60, and 90 days after sowing. And this screening was done for 3 years in the rainy. And for that, we went for mean versus stability analysis, that is GGB plot, to know which are the consistent genotypes, which are consistent along and across the years. So that we came to know that we have ICG 163 and ICG 10479 and ICG 111. These are the stable genotypes that are showing resistance to this disease. But even though we can't tell them they're highly resistant, but we have moderately resistant. And this is about the primary gene pool. And we have developed one more assay about known as oxalic acid assay. This is a lab-based assay. This was developed because the field screening is more laborious. And one more thing is we can't maintain the uniform infection in the field because this particular pathogen is not uniformly growing in the field. It is very erratic growth. It is having very too much erratic growth in the field. So this uniformity will make us like there is escape in disease, and we may enter into a genotype which is susceptible into resistant. So this particular protocol doesn't interfere any environmental influence. It will and this is based on oxalic acid as a principal agent that is produced by this particular pathogen. This particular pathogen, that is Sclerotium ralsii, produces maximum of 19 millimolar concentration considering all the races of this Croatian See, so we have used 20 millimolar concentration as the standard concentration to consider and millimolar control was used to check any genotype have like, high resistance or have save the cell so that the infection can be saved. But means the infection can be saved and the plant will be safe. And if the infection is too much, that means it is unable to control, then it will signal to autophagy. So that these are the genes, these red ones are the genes what we have identified through GWAS. These are involved in autophagy. That means they will signal to the plant for programmed cell death, so that it will sacrifice one of its cell which is infected to save the other cell, other neighboring cells. And after identifying all these markers, that is 13 genomic regions, we know that there will be the output of GWAS will be always the validation is must to come to final conclusion. So we validated using the same on 524 for genotypes. We went for allele mining. After that, we filtered for phenotype screening to come to conclusion that they are all for. We came to know that all these different population these 13 alleles. When we think about as a perspective of the breeder, if you have more number of alleles on different chromosomes, it will be difficult to transfer. But we already have few genotypes which are having the combination of alleles, so they can be used as a donor for this particular disease for moderate resistance. And this was the phenotype screening that was done using oxalic acid assay for these 8 genotypes. After this in silico validation, we went for validation using Cas markers, and this marker validation included breeding lines, land races, and even pre-breeding lines that were developed using secondary gene pool genotypes. And these 5 markers were validated, and they can be used in breeding program for marker assisted selection. And when we came to know about we have only moderate resistance in primary gene pool, that was very less when we come to like having a high resistant genotype. So we went for screening of secondary and tertiary gene pool. And when we screen for tertiary gene pool, that is Arasis glabrata, we came to know that we have a large variability in the 77 genotypes that have been maintained at ICRISAT by Gene Bank from 1991 as a live repositories. These have been maintained from 1991 by the ICRISAT Gene Bank in a glass house from from many years, that is from 1991, and we screened them. Yes. Then we came to know that after screening, we came to know that there are 13 genotypes resistant for this particular disease. And the crossing, so after out of 13 genotypes, we had one genotype that is similar, which is having structure of leaf structure and stem structure similar to the cultivated one. So we are using that in crossing program, and the crossing from tertiary gene pool is not so easy, and the crossing program is going on by using embryo rescue technique to derive lines, but we are trying it. We have not got the product out of it. And we can see the resistant genotypes here, that is the 8168 that is used in crossing is resistant to both LLS and stem rot. And after seeing the tertiary gene pool, we went for secondary gene pool, where we have developed 15. Yes. 1 minute. Yes, sir. Developed synthetics, and we have developed a population from synthetics by using different gene pools, from secondary gene pool using different species. And from that, we came to know that these ILs, which are that is 1, 12, and 24, the below ones have high resistance to this disease, and these are interrogation lines, and these can be directly incorporated into our breeding program as a source of resistance. And conclusion of this research are like we have developed a lab-based assay which can be used as an alternative or complementary to field screening when we have a large number of genotypes to screen, as it is difficult to screen in field. And we have identified different sources of resistance across different gene pools which are having different level of resistance. So these can be used as a donor for the breeding program. And these secondary these ILs developed from secondary gene pool are highly resistant, and they can be directly incorporated into the breeding program right now.
Thank you. Thank you. Thank you very much, Brenda. Thank you so much.
And these are the acknowledgements.
Thank you. Thank you very much. I'm sorry, we cannot, it's a very interesting topic. There may be many questions, but we cannot afford giving any additional time. Maybe those who have some questions can talk to Virendra directly. Now I invite the next speaker. Samir Pokhral is the one who will come to us, and he will talk on testing agile genetics in a complex multi-parental peanut population. Floor is yours, Samir.
All right, hey everyone, I'm Samir Pokhral. I'm a PhD student in University of Georgia. If you know Peggy Ozai-Sakins, I'm her student. So today I'm going to discuss about Peanut Pan Magic platform, putting agile genetics to the test. Okay. So agile genetics is basically a term coined by this paper by Justin Vaughan, Walid Korani, Josh Clemenger, and Peggy Ozai-Sakins. So basically, it is an idea of reaching to the single gene resolution level. We know that reaching to a single gene, it's quite a bit of job. It can, well, it's a tons of effort, and it can take over a decade to do that. So agile genetics basically relies on structural population, large-scale phenotyping, nested subsampling for the sanity test, exhaustive genotyping using the founder pangenome, and also refining the region with deeper sequencing. You can refer to this paper for more detailed information about the agile genetics. But if we look at the workflow of agile genetics, first we have the founder lines from which the high quality genome are extracted to develop into a pangenomic graph. So with that, the biparental or multiparental population need to be developed from the founder lines. That basically has two different ways of evaluation. One is on the unreplicated plant. We are talking about a really big scale, around 5,000 to 10,000, a single plant evaluation at a field level, and direct sampling in the field. Whereas the other part would be the nested subsampling from the single plant evaluation and then replication, and using the pangenome graph, bunch of evaluation for the sanity testing. So third part would be genotyping and the bioinformatic side of it, where the individual that we subsample needs to go for a skin-seq, which is a low-pass whole genome sequencing, and since we know what samples are those, we can actually develop some modeling to test the population structure and things like that using the pangenomic graph. And also the other part is direct, as I said, direct sampling in the field and depth enrichment based on what we find through one way of pangenomic graph, and use that We use that finding to enrich the depth using amplicon-based sequencer. We call it iterative depth. Again, you can refer to the paper for detail, and finally reach to the candidate gene level. So this study is actually a simulation, the paper that I just discussed about, which has not been tested, and I'm trying to test that in a PanMagic platform. So again, PanMagic refers to the pan genome, and magic refers to the multiparental population that we have. And talking about the first component that we are testing to test the agile genetics is using the founder line to develop the pan-genome graph. For that, we have this founder line that we used for magic population development. So we have 18 different divergent and agronomically important founder lines that we developed into a magic population. So idea behind this is we all work with peanuts, so most of us would know these lines or genotypes have different traits that would be desirable. Some of them are drought tolerant, stem rot. We have quite a bit of talk on stem rot resistant, high early content, high yield, and several other traits. So idea behind this the magic population is that we could actually get the traits, several traits incorporated into the single population or individual, single individual level. For that, we had this team of magic population development. First, we made the biparental cross among all the 18 divergent underline that I just discussed. So after making the biparental cross, we reached to the 2A F1 level. The 2A F1 would have two parental genome present in its pedigree. And that 2A F1, the crosses were made among themselves in a half diallelic design to develop a 4-way F1. Now the 4-way F1 would have the mosaic of 4 genomes present in its pedigree from the parent. And then again, we would make the cross between the 4-way F1 to develop a 8-way F1. So 8-way F1 will have 8 genome of parent present in the pedigree, followed by the 16-way F1. So basically, although we have 16-way F1, there is several different studies going on with that. And my study is particularly focused on 8-way F1. Again, I just wanted to focus that it started from 18 parental genome, and 8-way population would have 8 out of 18 genomes in it, and it won't be balanced in terms of 8-way population. So taking that founder lines, those founder lines that I just discussed about, and using the TFRunner as a community reference genome that we use, I tried to develop the pan-genome graph. So that started with genome construction first. This is basically the pipeline I use for the genome construction. We use this PacBio hi-fi sequencing technology, which is a long-read sequencing technology and very less error. So it's pretty desirable, and with a 20x coverage. And that would give us raw sequence, which is already pretty awesome and didn't need to do much in terms of filtering and others, but I filtered the adapters using this hi-fi adapter field software, and assembled the genome using hi-fi ASM. So assembly was 98% complete, and N50 value, as you can see, is 45 MB. So pretty complete and contiguous assembly was delivered using the hi-fi ASM. Again, so altogether 19 genome, 18 of the founder lines, and 1 TFRunner reference genome. And finally, I scaffold the assemblies using the ragtag scaffold software with the progenitor reference genome for a pseudomolecule level. So, I mean, we all work with peanuts, so probably one of the challenge that we always get in terms of genome assembly in a tetraploid like peanut would be subgenomic collapse. So subgenomes of peanut are quite similar. So when we try to assemble the genome, it's likely sometime that some of the region would be mismatched, and we could see tons of subgenomic collapse. But the pipeline we used has really reduced subgenomic collapse. So before, we just tweaked some parameters in the HiFi ASM software to test that, and it's been really reduced by 6-fold. This is just a case of C9NR genome. So word of caution, if we are assembling the genome, it's always advised to look for subgenomic collapse. And finally, using the scaffold developed from the RAGTag, I developed the pangenome graph using the minigraph actor software with total SNP of 2 million SNP and indels 1.3 million, and around 4,600 indels were more than 10 kb. So that's a really big insertion region that we could see in the graph. And the beauty of pangenome, we can actually see the genome. This is the MOV aligner, and these are I don't know if I can see the cursor, but so these genomes are okay. Let me try to. Anyway, so if you see the highlighted regions in the MOV aligner, you can see some of the genomes have the insertion region, some doesn't, and that's pretty powerful software. If we zoom in a little bit, thank you. So right here, these are different genomes. I had 18, it's just a snip. It's just a image showing some of them, and you can see some of the genomes have the region and some doesn't. And if we zoom in further with sequence 2 map software, you can actually see the difference at the nucleotide level. So, I mean, it's a pretty powerful tool, and with that, we'd be ready to actually map the traits. And this is the variant distribution heat map. If you look at 20 chromosome of peanuts, several different SNPs, as I talked about, was present, and some of the interesting chromosome, for example, chromosome 9 had really high SNP, probably due to the presence of cardinessa introgression and TFNB high olic cultivar that we have. Similarly, chromosome 3, we have ISC 322 that might have the cardinessa introgression trait. So basically, the idea is that we have this SNP platform already developed, so we need to phenotype and genotype the traits of interest, and we can map the traits. That takes me to the second part, which is phenotyping component of it. So Agile genetics, as I said, is based on large single plant phenotyping. So we had this 8-wave population when I joined my PhD, and then we advanced them to the F2 generations. At the F2 generation, we actually conducted the study that we wanted to conduct. One was on the drought, and another was on the white mold. So white mold or stem rot, Birendra Kumar already talked much about it. I'm not going much detail to that, but so we select the progeny having stem rot resistant parent in their pedigree at the F2 generation. And again, for the drought, we selected the progenies that had drought tolerant parent present in their pedigree for the study. So we evaluated 900 single plant at F2 and F4 level for 2 years for the drought tolerance using this structure called Renaud shelter. And same thing for the white mold, 700 individual of F3 and F4 for 2 years. And again, not going much detail to that, but I would like to say, so this is how the plot would look like from an aerial drone image. So we would leave the plant to grow in a normal condition, and 70 days before the harvest, we would pull the structure that is Renaud shelter, and that would provide rest to the plant, and based on which we would evaluate the plant performance. So this is a scale that I rated the drought for. It goes from 1 through 5, 1 being the single plant that didn't have the any drought symptom, and 5 is the plant that's actually killed by the drought, and 2, 3, and 4 are intermediate, successively rating for drought. Again, these are F2 and F4, but we can still, if I look at by the family distribution at F1 level, we can see that there is a strong genotypic effect going on. And these are the checks, all the blue ones and all the gray ones are my actual experimental family that I'm testing. So just letting you guys know that we have a strong genotypic effect going on. And as a word of caution, I would also like to say that if you are doing a drought experiment in the field, there is always a chance of field effect. So especially these age rows 1 and 2 and 9 and 10 which are lighter in colors means they have the lesser rating, means they are less stressed than the inner rows going through 3 to 8, which have high stress. And again, the volumetric water moisture outside than inside, even inside 1 and 5, which are the 2 age beds, has higher water moisture than 2, 3, 4. Just to be careful about something. And again, the second trait that I discussed was the white mold or the stem rot disease. For that, it's a little bit different than what previous speaker talked about. So we have this Georgia 12Y canopy planted in a 15-foot plot, and 5 experimental material were planted at each flag, and we went to the field and infected the experimental material using the Athelia rhodesi fungus and set it for 3 weeks and start rating them on the scale of 1 through 5 again, 1 being the best, no apparent symptom, and 5, the plant being killed with the white mold disease stress. Again, we are just showing that we have some family effect going on, a strong genetic effect going on, and I think we'll have the phenotype is promising in that sense too. But we went a step further. So after I did the drought study in 2023 and white mold study in 2023, I selected the line that has tolerance to both disease. And then we sent it to Puerto Rico for advancement, and then followed by stress induction, a few more years, and selection is ongoing so that we could release several germplasm, including the 8-way population that we would like to release as germplasm. And the last part of the agile genetics would be the genotyping, where we have genotype 4200 progeny for 2 years using the whole genome low pass scheme-seq technique. It's a 1x coverage technique, pretty cheap method, and genotyping is ongoing in Hodgson Alpha, and they're doing pretty awesome job in terms of using this technique. And I'm hopeful that I'm gonna get good results out of that. So in summary, agile genetics relies on large population, single plant phenotyping, and exhaustive haplotype knowledge. The component of agile genetics I'm trying to test in the magic population that we have developed, and Pangenome would facilitate that. And we evaluate the older generation progenies for complex red, drought, and white mold, and phenotyping is promising and genotype is ongoing. And please stay tuned for the genetic dissection of the trait. With that, I would like to thank my advisors, Justin, Peggy, Josh, Dr. Brenman and Dr. Holbrook, awesome people to work with, and Jason Shannon, USDA, NIFA grant, Hudson Alpha for all the genotyping work, and POA lab, OJS Circuits lab in Tifton for all their help and support that they have been doing with the project. And finally, AAGB 2025 for the opportunity to let me talk here. And I think I'm ready to take any questions you have. Thank you.
Thank you. Thank you very much, Samir. There's the possibility of having one question. Yes, Shruti.
Samir, congratulations on your wonderful presentation. My question is, when you go for multi-parental crossing, did you see any linkage drag for the negative trait alongside the target of interest, and how did you overcome it?
I didn't do that part of the project. When I started, it was already well established and it was at the A12 level. But I can reach out to the people who did and answer to that question.
what is the geographic representation of your 18 parents? Is it only from the Georgia or US area, or it's representing from Africa as well as from Asia?
It's mostly from US itself, but we have some ICG line. I guess it's pretty popular in here too. But mostly it's mostly based on the US.
Well, thank you. Thank you very much.
Thank you very much.
We may have more interactions here. So we have our last speaker of the day. Last speaker is Dr. Jianping Wang from the University of Florida. We have him online. Is it online or recorded one? Online, recorded. Yeah. And Jianping will be talking on understanding the molecular mechanisms on root nodulations in peanuts.
This opportunity to present the research currently going on in our lab about the IP mutagenesis and the effort towards IP allele discovery in peanuts. So as you all know that the crop traits are determined by the DNA sequence, such as the plant height, the fruit size, and the disease resistance, which we have been heavily working on right now. They're all under the control of the DNA sequence. On the other side, however, the epigenetic marks or the IP marks are also very important for the crop trait determination. So the IP marks are the chemical tags, such as the methyl group or the ethyl groups added to the DNA molecule, or the histones interacting with the DNA molecule that can infect the DNA sequences. So that IP marks can turn off the gene expression. Depends on what kind of IP marks. They can turn off the gene expression or turn on the gene expression. Some of the environmental factors, such as the drought, the temperature, or the soil nutrients, that can interact with the IP marks to modify the gene expression, therefore to impact the trait. So many studies right now have shown that the IP marks can help the plant to adapt to the environmental stresses, or can influence the plant yield, the growth rate, or the nutrient utilization efficiency. And the DNA methylation is one of those IP marks. It's very important. It occurred mainly on the cytosine in the DNA molecule. Mainly it's after the DNA synthesis. So the cytosine here, the methyl group can be added to modify the cytosine in the newly synthesized DNA strand through the DNA methyltransferase. And the methyl groups can be added, that is the DNA hypermethylation process, or can be removed, that is the DNA hypomethylation process called unmethylated DNA. And the DNA methylation can occur at 3 different contexts, include the CG, CHG, or CHH, different contexts. For the CG context, normally it's in the transposons or the repetitive DNA sequences. So the C-methylation involved in the gene balancing. And in the CHG context, the methylation basically is reinforced the repression of the DNA to protect the genome integrity. And for the CHH context, normally the methylation is more dynamic, it goes or comes. It mainly responds to the environmental changes. So overall, the demethylation is very important for the genome integrity, the chromosome stability, and also it's important for the crop to adapt to the environmental changes. For example, here, if the DNA there is a gene structure, if the promoter region is methylated, it can turn off the gene, cause the transcription stops. That is could be response to a certain environmental signal. So the IP mutagenesis is a process of inducing the random IP mark or epigenetic changes that can be heritable and impact the gene expression without really changing the DNA sequences. Some of the environmental factors, for example, the tissue culture in the high plant hormone level, or different chemicals or radiations or extreme temperatures can all cause the epimutagenesis. And some genetic factors, for example, the wide distance hybridization or gene mutation, for example, the genes involved in the DNA methylation or the histone modification, if those type of genes get mutated, it can also cause the epimutagenesis. So here I would like to introduce a chemical that can cytidine. So the 5-alpha-cytidine is an analog of the cytosine. Here is the cytosine base. You can see the structure is very similar between these two, cytosine and the 5-alpha-cytidine. So the cytosine has a H group here at the 5' carbon, but the other cytidine does not have that H atom. So for the cytosine to be methylated is a methyltransferase at the CH3, the methyl groups, to the 5', and with the 5' other cytidine, the methyltransferase cannot add the methyl groups to the 5'. So that basically causes the demethylation. So if this other cytidine is incorporated into the DNA molecule during the DNA synthesis, then the methylation cannot happen most likely and cause demethylation. So this chemical has recently been used as a DNA methylation inhibitor to study the DNA epigenetic landscape. In our study, we used the 5-alpha-cytidine to treat the peanut seed, pea browner, and we're very excited to see even at the M0 generation, we can see a lot of morphological changes. And this actually was done by an undergrad student in our lab several years ago. So our question is how stable are these variations? Or are these traits can be really inherited across different generations? So we grow the seeds from the M0 into the M1 generation in the field, and we can see the morphological variations are still there, which is good. And we also evaluated the part traits, and we found the one of the mutants, actually the seed size is much bigger than the pea runner, the control. And we grow the M1 seeds into M2 generation in the field, and we can see most of the lines are still segregating, not uniform. So here is a plot. One plot is from one plant seed one and one plant seed. And interestingly, one of the plots showing very uniform bundle structure in terms of the morphological architecture. So we're very interested in this line, and we grow them into the M3 generation. They're still consistent. So in the M4 generation, we start to phenotype this mutant. We call it a dwarf plant, a dwarf IP mutant in the field. And here in the field, if you can update, it's very tiny compared with the wild type, which is tip runner. You probably know how the tip runner grow in the field. It's a runner type. And we measured the canopy, the main stem, the growth, and also the leaf length with petiole length, the main stem length, and the root fresh weight and the length, and also overall the biomass. And from here, you can see the epimutant. The stem length is much shorter than the wild type, and it grows relatively slower. And in terms of the canopy, the mutant is also much smaller and grow much slower as well than the wild type. For all the other phenotypes, all the other parameters we assessed, particularly the plant fresh weight, we can see the wild type have much bigger plant fresh, more plant fresh weight than the IP mutant. So we're wondering whether we can map the IP allele controlling this dwarf trait. And we made the crosses to develop the F2 mapping population between the IP mutant and the wild type. Did the reciprocal crosses as well. And here is the F2 populations in the field. And because the IP mutant is dwarf, it's basically embedded in the plot with all of the wild type. You have to take them out to be able to phenotype them clearly. And here is a summary of the F2 population, the segregation situation for the population, which has the dwarf as a female parent and the wild type as a male parent. For this population, we had 373 individuals, include 81 dwarf and the 292, which are normal individuals. And we did the category test, and we found out it's following the normal to 3:1 ratio. F2 population with as a female parent, the dwarf as a male parent, we have two. And this population, there's a lot of to follow up to study on that. So we're wondering whether we can map the IP allele. So we borrowed the idea of the QTL-seq or the BS-seq method to develop our own BS meso-seq protocol. And here I will quickly describe the QTL-seq or BS-seq method. Basically, you have a biparental lines which are have very constructive traits, and you have the F2 segregating population, segregating in terms of the trait you are interested, and the distribution. You have the lower the individuals showing that trait, and the higher the individuals showing the higher traits, and you bulk those lowest and the highest individuals into two different bulks, and then you sequence the whole genome of those two bulks, and then you call the SNP. When you get the SNPs, you compare your SNPs with the reference or the parents, you get the SNP index for both of the bulk, and you subtract the SNP index of one from the other, you get the delta SNP index, and then you plot that and find the candidate regions, and then within the candidate region, you identify the SNP or the gene with the SNPs, and then you annotate them for the candidate gene. And similarly, for the BS mesocytic, we had two different bulks, the dwarf plant bulk and the normal plant bulk from the F2 population. BD is the dwarf and the BN is the normal plant bulk. And plus the dwarf and the wild type parents, we did the whole genome bisulfite sequencing instead of just the whole genome sequencing. So for the whole genome bisulfite sequencing, we have the DNA extracted from those bulks, and then we converted. We did the bisulfite treatment to convert the unmethylated C to U for the downstream sequencing, and later on you can identify which C were methylated, which C were not methylated, and then you can call the methylation level of those Cs, and then you get the methyl index of those two bulks, and then you subtract this one from this bulk, you get the delta methyl index, and you plot the delta methyl index, you get the candidate region, and then you can identify the methylation or the epi alleles contributing to this contrasted phenotype in those two bulks. So we did the methylation analysis of those two print lines, the mutant and the wild type. And here is the overall the methylation at those three different contexts. And here is the epi mutant. You can see the epi mutant compare with the wild type. The methylation level is significantly lower than the wild type in all of those three contexts, which means our treatment for the demethylation is successful. And that methylation, we scanned the whole chromosome, and here is a representative of the whole chromosome wide the methylation level comparison at those three different contexts. And this is a mutant, and here is a wild type. You can see the mutant, epi mutant, across the whole chromosome had a much lower methylation level compared with the wild type in all three different contexts. And we also identified the differentially methylated regions between the epi mutant and the wild type. And here we can see the CHH DMRs are more abundant than the CHG and the CG context. And mostly the DMRs are hypo DMRs, which means the epi mutant have less mutant, have less methylation. Of course, that means our demethylation treatment with 5 other Z was successful. We also noticed some hypermethylation, hyper DMRs with all three contexts, which we don't know what caused that. It could be the hypo DMRs could impact the nearby region and cause hypermethylation. We don't know a lot of to continue to study. And the DMRs, we found the CHH DMRs the region typically is longer than the CHG DMRs, which is smaller, and here is a CG DMR length. Okay, so according to the CS meso-seq pipelines we developed in our lab, and we scanned the whole chromosome and identified the chromosome 15 at the beginning of the chromosome 15. There is a very significant signal to indicate between the two bulks. They're dramatically different in terms of the methylation level. So we're going to investigate what the methylation changes in this region, and this project is still going on. And the take-home message here is that IP mutagenesis can generate a very stable IP mutant without any problem, and this can be a very powerful tool to increase the DNA.
Thank you very much. You can still continue reading it. In fact, this was a wonderful session, very diverse session from heat tolerance, allergens, to shell strength, to diseases, and of course to epigenetics. Wonderful, very informative. Just not to proceed further, I will request my co-chair for his comments.
Thank you, Kuldeep. I think very nice, excellent presentations. Kuldeep said, very diverse. I think a lot of information, a lot of discoveries, we listen, a lot of marker discoveries, polymorphisms, marker trait associations, donors identified, mutants, developed. Probably just to close, as a comment, I'll just start by asking questions. So what? Probably, should I ask it in a different way? Where from here, all this information need to be utilized to develop products for target markets. Do we have some of the markers, an in-house genotyping platform or commercial genotyping platform where, the breeders can access and utilize them for developing products? I think this is related to what Dr. Bera commented yesterday. We have a lot of QTL discoveries, marker trait associations, but we have very limited markers that could be applied for breeding except leaf spot and hyolic. So it would be, important if we also focus in such areas on the way forward. That's just my brief comment. Thanks again for the excellent presentations. Good job.
Ladies and gentlemen, uncertainties are unpredictable. So was our morning's whole howl. With a strong leadership and team's willingness to tackle collectively sets the stage for extraordinary. With this takeaway message, we'll close this session, thanking all the speakers, chair and co-chair conducting this session. We'll now break for tea. Sharply by 11:10, we will gather here and begin the next session. I'm giving only 5 minutes break. Please, sorry, but I have to keep up that time. Everybody will gather here within 5 minutes. And yes, please get your coffee and we'll kickstart the session. Thank you.
Hello. So the next session is the next generation researchers, and I
this higher seed size gives rise to higher yield in both the location at Trombe and Gauribindu that we tested it for. And then we wanted to know what is the reason behind this increase in cell size, because being a polygenic trait, mutation would have knock out one or two things, and we could stumble upon the gene which is having major effect on it. So for histological characterization of mutant, we harvested the pods at different days after flowering, and we understood that all the pods of mutant TG89 were having higher quarterly tolerance size at all the 5 growth stages of the pod development. When we checked the cell size and cell number using electron, this environmental scanning electron microscopy, we found that the cell size was higher for mutant in all the growth stages than the parent. However, the cell number was insignificantly similar with respect to mutant, leading to the conclusion that the increase in the cell size of increase in the quarterly tolerance size of the mutant was attributed to the increase in cell size and not the cell number. So to understand the genes behind the possible reason for this, we cross this mutant with a distant genotype ICGV 1507. Since it was a mutant, we could not directly cross with the parent. We didn't get a lot of polymorphism in parent mutant combination, so we chose a distant parent, and we did a bulk segment analysis choosing in F2. We got a very good distribution of seed size leading to its genetic in nature. We chose 2 bulks and did bulk segregation analysis, wherein 3 markers could positively distinguish the 2 bulks from each other. And these markers belong to A05 chromosome, making our focus shift on to that chromosome. Yet we also did SNP analysis by DD-RAD-seq, some SSR and mite marker analysis, and we found that 4 QTLs were identified on A05, A02, B010, and B01 chromosome. Out of these 4, 2 were major QTL explaining more than 12% of phenotypic variation, that is A05 and A02 common. When we saw the allelic distribution of these particular QTL flanking markers, we found that the markers on A05 indicated that the gene contribution by mutant was responsible for the positive additive effect for this QTL, whereas the second major QTL A02 indicated that the QTL was derived from the ICGV9 type of parent. So our focus again came back to A05 chromosome of the marker SNP 05 103 14,987 and TE 470. When we look for the physical distance between the physical distance that it mapped came to about 4.0 MB region, which harbored around 100 genes. We did the analysis of these 100 genes and narrowed down few genes which would be related to seed size, and we narrowed down to AH5M7JWE, which had a role which had a decreased expression in mutant than the parent. This mutant had a homology in BS1, big seeds 1 gene of soybean, which is known reported gene for seed size regulation in soybean and glycine max. So we thought there are 29 such big seeds kind of genes in groundnut. We analyzed expression level of all of them. 3 of them had lower expression, and 1 of them had a higher expression than the parent. And this data, we also did a transcriptome analysis wherein they indicated that the genes for circadian rhythm and hormonal biosynthesis were upregulated in mutant. And this data also indicated, this was confirmed with RT-PCR analysis of 12 DG. So mechanism, the putative mechanism that we deduced from this, that TFI, the big seed 1 gene of groundnut mutant is a transcriptional repressor. It sits upon the genes responsible for circadian rhythm and hormonal biosynthesis. So when this in mutant, this gene is knocked down, this repression is elevated, and this TFI complex, because of lack of TFI protein in it, gets broken down by ubiquitination pathway, and the repression on downstream processes of circadian rhythm and hormonal biosynthesis elevated, and the seed size, there's an increase in seed size. So we synthesize a specific marker for this and utilize this marker for breeding and selection for last seed size. We could identify 12 lines.
To wrap up time, so you have.
Yeah, it's done. So we utilize this marker flanking QTL1 and isolated 12 breeding lines having high yield as well as large seed size. Thank you.
Thank you, Ms. Poonam. I request Ms. Rati Satish Chunletia to present on identification of homolog DOG1 within a major QTL region of seed dormancy in groundnut.
Namaste and good morning to one and all. So I'm Rati Satish Chunletia and let's start this presentation as a story. So yesterday, Vera sir and Kuldeep Singh sir both had focused on the change in climate which we are facing right now. And from past 3-4 years, we are having rainfalls during the harvest. So because of this erratic change in the rainfall patterns, it is difficult for farmers. So the problem is this in situ germination. So what happens is the pods will grow inside and this product now is not useful for the farmer because we know it is not going to help out with seed production nor can we germinate it in the next season, no oil production. So this is the problem. Now the solution, we already have a solution that is the dormancy when we introduce dormancy in our seedlines. We had it previously, but the problem was it got lost during domestication. So the objective of work was to find a region of 14 to 21 days dormancy period so that the farmer can harvest it and during that and not more because then they have to re-sow it. So we started with two parents, TG51, which is a relatively new variety which is non-dormant, and we crossed it with TG22, which is a dormant variety and relatively older one. And a real population was constructed for 160 lines, with 160 lines, and it was mapped with almost 235 markers. And the phenotypic data here used for this mapping was days to germination. So we started from 0 day to when we put it for germination plate testing, and we ended it till 45 days. That is the TG22, the dormant parent whose dormancy breaks at 45 days. Further, now with this map, we can see that we got a 5 centimorgan difference, and the QTL was mapped between T373 and 202. So because it was a wider region, we went for BSSEC, and with the BSSEC also, it was not very conclusive because as we can see here, the whole chromosome was associated positively with the trait. So we use this BSSEC data to construct some insertion deletion markers, and they were then added to the map for the saturation. And now we got a new QTL between indel 53 and 43, which reduced the centimorgan distance to 1.84 and a physical distance of 10 megabase pairs. Now, side by side, we had derived some nils line from the real population, and these nils were sent for transcriptome. And from transcriptome data, we got 47 common upregulated genes, DGs, and 53 common downregulated. Now, these DGs then were taken for validation through qRT-PCR, and 5 common upregulated and 5 common downregulated genes were used. Now, these genes were validated, and from here, we had looked for our the course mapping QTL region that is from 62 megabase pair to 108 megabase pair, and only those genes from A04 which we got from our mapping were shortlisted, and 3 of them were tested with qPCR, that is magnesium transporter, ubiquitin, and transcription factor. This was further checked for 2 time points, that is 104 days after sowing and 114 days, which was also the day of harvesting. And we can see that it was upregulated in both the time points. So till here, this is our finding. The key finding is that we got this TGA7, which is a homologue to DOG1, and it may be responsible for our dormancy trait. Now, the future aspects that we are going to cover for this is that we are going to go for functional analysis for DOG1. We are going to try and make some snap markers or cast markers to even saturate and pinpoint the location. And plus, apart from that, we are going to test this DOG1 till the dormancy breaks. So these are the key findings and the references. Thank you.
Hello, thank you, Ms. Rati. I request Ms. Lopamudra Giri to present characterization of Nothospora personata causing the late leaf spot disease in peanut across the major peanut growing regions in India.
A warm good morning to one and all present, to all the dignitaries on the dais and off the dais. This is Lopamudra Giri, a PhD scholar from Department of Groundnut Pathology, ICRISAT. And today I would like to present a small part of my PhD work, that is the characterization of Northopacillora personata causing late leaf spot of groundnut from major groundnut growing regions of India. So we all know that peanut is an important legume globally, serving as an food and oilseed source for millions of people. But the peanut crop is there are many challenges faced by peanut crop. There are biotic and abiotic stresses, and among the biotic stresses, late leaf spot pathogen caused by Northopacillora personata is one of the most devastating foliar pathogen, whereas the yield loss is seen up to 40 to 90% in this infection. So since it is a foliar pathogen and it is highly adaptable and evolving pathogen, which can rapidly acclimatize to new environmental conditions, and for this purpose, monitoring and prevalence as well as genetic diversity of this pathogen in various regions is a requisite in tracking its spread and identifying the highly risk areas. So my study has mainly focused on the study to evaluate the disease severity as well as molecular variability of Northopacillora personata across peanut growing states of India. So at first, we have collected almost highly infected leaves with LLS from from 5 different states of India, that is Odisha, Andhra Pradesh, Tamil Nadu, Telangana, and Gujarat. Whereas the spores of these isolates were collected with the help of cyclone spore collector and stored at -20 degrees Celsius. And for pathogenicity test of this isolate, the spore suspension of concentration, that is 5 into 10 to the power 4, was prepared from the spores collected from different states. And in order to carry on the pathogenicity test, the susceptible variety TMV2 is used. 45 days old leaves were collected from TMV2 variety and used for pathogenicity test. Here you can see the pathogenicity test of the highly virulent isolates have been depicted, each representing each state of the India. So not much, but slight variation in the pathogenicity test. Like almost all the isolates were found positive for pathogenicity test, and few were highly virulent. And there were little bit difference in that symptoms appeared, and the shape as well as diameter of the leaf spots. Whereas when coming to morphological characters, almost you can see that in the image, you can see the conidiophore bearing conidia, and each conidia has been measured, whereas the conidial size has been also recorded. And there you can see just slight variation in the septation, like few are 3, 4, and 6 to 5. And almost all the conidia are cylindrical to oblicate in shape. After that, molecular characterization of these isolates was also done. DNA of the spores was isolated by using amine kit, whereas specific primers like RPB2 forward and reverse has been used, and amplicon size was found to be approximately 800 to 900 base pairs. So you can see after the sequencing results, we have analyzed our data, and with the help of MegaX software, a phylogenetic tree was created. And here you can see that all the isolates have been formed 3 clades, whereas our isolate from Andhra Pradesh is more similar to Iran and Netherlands isolate, whereas our isolate from ICRISAT Telangana is similar to Argentina, whereas other isolates are similar to Iran and Argentina isolates. So the future line of work which we are going to carry on, like by characterization of this pathogen, we can identify the pathogenic variability, which can help us to develop the efficient as well as region-specific management of the management strategies against these diseases. Whereas, moreover, whole genome sequencing of this isolate could identify the key pathogenic related genes, paving a way to innovative control measures and development of parent varieties with enhanced stable resistance. Not only this, but also the work on biocontrol management of this disease is also in the pipeline. So thank you everyone for your patience listening.
Thank you, Lopamudra, for finishing well in time. Now, next presentation is online presentation from Dr. Xin Wang from OCRI, China. So she will be talking about the genetic analysis on cold tolerance in groundnut.
increasing granite production. Obtained no and temperature has adverse effects on granite growth. Thus, improving trajectories, analysis of genetic diversity and areas, discovery of yields for enhancing granites, and optimizing PI of this project. And Professor this background is the we applied and successfully obtained an international cooperative
So this is 5-minute talk, but the technical
expansion of planting areas to high latitude and altitude regions is necessary for increasing granite production. No temperature has adverse effects on granite growth. Thus, improving cold tolerance has great significance for expanding planting areas, ensuring yields, enhancing quality, and optimizing supply chains of granites. Under this background, we applied and successfully obtained an international cooperative project with Ecriset last year. The project title is Analysis of Genetic Diversity and Discovery of Functional Genes for Cold Tolerance in Granites. I am the PI of this project, and Professor Janina is the co-PI. Progress 1. The cold tolerance was significantly distinct among different germplasm. For example, some cultivars have a high seed germination rates and high plant height and more pod lumber when they grow under cold, stressful conditions. Furthermore, some wild arches species stronger cold tolerance than the cultivars. The arches result trees and serve to survive winter. Unresolved scientific questions related to the cold tolerance was that the candidate genes or QTLs controlling this important trait has not been found or cloned. Therefore, in this part, a real population with 807 lines was developed using the two parents, and each YEF12 is sensitive one. Then, a high-density SNP-based genetic linkage map was constructed by genome resequencing. And finally, a major QTL controlling seed germination and cold stress was mapped on chromosome B9 with a region about 200 kilobase in size. For discovery and functional analysis of more cold-tolerant genes in granite An efficient platform for screening cotolerance genes was constructed by using yeast expression system. A total of 157 cotolerance candidate genes were discovered in granite using this platform. One candidate gene was proved to encode a flavonoid azane. Overexpression of this gene improved cotolerance by scavenging excessive rows in Arabidopsis. In future, we will cooperate with ICRISAT to perform research on the following three aspects. The first one is comprehensive evaluation of representative granite germplasm and discovery of candidate genes for cotolerance through GWAS and multiomics. And the second one is many special candidate genes for cotolerance in wild algae species. And the third one is developing gene-specific markers, discovering and creating novel materials with improved cotolerance. Okay, that's all. Thank you.
Thank you, Dr. Shin Wong. Now I request Ram Raju Manasa from Mangaru. She will be talking about the biochar application in sustainable approach for managing the stem rot disease in peanut caused by Sclerotium rhopsi.
A sustainable approach for managing the stem rot disease in peanut caused by Sclerotium rhopsi. Stem rot is caused by Sclerotium rhopsi, which causes the yield loss of about 80% in severe cases. Chemical control is not a sustainable, and the resistant varieties are not up to developed. So prompting for the alternate approach. So we have used the biochar. Here the biochar is up to, we have prepared from the peanut shells. At the temperature of 450 to around 450 to 500 degrees centigrade through slow pyrolysis. This table represents the basic properties of the peanut shell biochar, where the biochar is prepared from the peanut shell at the temperature of 450 to 500 degrees centigrade, and the pH, it has a pH of 9.54 with an OC of 12.34%. It also has a good soil aeration, water holding capacity, and porosity. It is rich in the macro and micronutrients. These properties made the biochar. These properties has made the plant. It has developed the potential for the plant health development. Apart from that, it even increased the suppressive nature of the suppressive nature against the pathogen. Here we have evaluated the different concentrations of biochar against the stem rot disease. Here, 3 experiments were conducted, one at laboratory conditions, glasshouse condition, and field conditions. In glasshouse conditions, different genotypes, 4 different genotypes were used. They are ICGV 1710-02, 1810-35, 2110-2111-07, 1710-25, and with the checks, that is CS319 and TMB2. Here, under glasshouse conditions, the biochar has been applied to the soil 2 days prior to sowing, and sowing has been carried out. After 30 days after sowing, inoculation has been done, and at every 15 days after inoculation, the % disease incidence was calculated. In case of field conditions, the biochar was applied to the soil 30 days prior to sowing, and the sowing was carried out. And after 30 days after sowing, the inoculation has been done. And for every 30 days after inoculation, the % disease incidence was noted. This is the same analysis of the biochar, where the results depicted that an increased prevalence of interconnected micropods and suggested that there will be increase in the biochar porosity. Under laboratory conditions, we have tested the different concentration of biochar against the stem rot pathogen, where the results reveal that the biochar did not affect the growth of the pathogen at any concentrations when compared to the control, but upon closer observations, it significantly affected the production of sclerotial bodies when compared with the control, that is at 5% concentration. And under glasshouse conditions, this stem rot symptoms, it has shown that in inoculated control, the symptoms has been shown after 7 days post-inoculation, while in case of the biochar treated pots, the symptoms were delayed 10 days post-inoculation. And among the treatments, if we see that is at 1%, 3%, 5% concentration, the biochar treatment at 5% concentration has shown the reduced disease progression when compared with the other treatments. And among the genotypes, if we take the genotype ICGV 171002 and 181035 has shown the less disease production, disease incidence at the range of 20 to 30%. This is the performance of different concentrations of peanut shell biochar in the selected ABLs. This one is, first image is at the genotype ICGV 171002 and next one is 181035. These are the next, third one is CS319 and fourth one is TMB2. Similar results were obtained under field conditions. At 5% concentration, the disease incidence was reduced and it was shown at a range of 20 to 30%. And at 5% concentration, the population of Sclerotian ralsei has been reduced. And this is the pictorial representation of the biochar. When applied to the soil, there will be change in the soil characteristics, and there will be increase in the microbial population, and the mechanism of micropopulation has been increased. So the resistance of the plant can be increased when biochar was applied. Thank you.
Now I request Ms. Hima Bindu Badragiri, and she will be talking about the multi-locus genome-wide association analysis uncovers candidate genes for early leaf spot and rust resistance in groundnut.
Esteemed scientists, respected seniors, and my dear friends and juniors, a very warm morning to all of you. Here I am, Himabindu, a PhD scholar from OUAT, is before you to give a small flash talk on the topic. Multi-locus genome-wide association study uncovers candidate genes for early leaf spot and rust resistance in groundnut. Groundnut is an important oilseed crop originated in South America and is grown in several other countries like India, China, Argentina, Sudan, West Africa, etc. And in India, it is majorly grown in the states like Gujarat, Andhra Pradesh, Maharashtra, Karnataka, and some parts of Odisha. The most limiting factor for groundnut production is the biotic stress, that is foliar fungal diseases, which cause an yield loss of about 70%. So most of the farmers prefer chemical treatment for their control. But considering the environmental and the cost factors, it is better to use the disease-resistant varieties in the breeding, which are developed in the breeding program. So keeping this in mind,
your slides are not moving.
Yes.
Oh, it didn't move.
Keeping this in mind, we have sought to find the genomic regions which are responsible for the disease resistance. Early leaf spot is caused by Cercospora arachidicola and rust is caused by Puccinia arachidis. For both the diseases, high humidity is favorable and the temperature of around 20 to 30 degrees is favorable for disease progression. In order to find the genomic regions responsible for resistance, we have used 184 mini-core lines, which were selected from the 300 reference set. And we have used phenotypic data from 3 different locations for early leaf spot and 3 seasons data for rust. And we have seen that there are 2 resistant lines which are both resistant for early leaf spot as well as rust. They are ICG 11088 and ICG 11406. And using the whole genome resequencing genotypic data and the phenotypic data which we have got from 3 different locations and 3 different seasons, we have done genome-wide association study, and we have got 5 genomic regions associated with early leaf spot resistance and 12 genomic regions associated with rust resistance. And further, after finding the marker trait associations, we have gone for candidate gene discovery also. And few best candidate genes we have found are callose synthase, which is responsible for callose production, and sterol synthase, which is used in the production of brassinosteroids, which have a role in salicylic acid pathway. Of the identified SNPs, we have selected 4 best SNPs, which are clearly differentiating between the resistance and susceptible lines. And those SNPs we have validated on the Intertech platform, and we have found that 2 best SNPs are validated in different other diverse populations like pre-breeding lines, reference set, etc. And based on the genes which I have got, the candidate genes which I have got, I have made a hypothetical pathway where where the genes are upregulated in the resistant line and downregulated in the susceptible line. The resistant and susceptible line which we used are GPBD4 and JL24. These 2 SNPs which are validated can be used in the breeding program for marker-assisted selection. We are also doing work on exploring novel resistant sources of resistance to late leaf spot in groundnut. So far, GPBD4, which is derived from Arachis cardanaceae, is the only resistant source for LLS. But while we were doing allele mining using the markers derived from GPBD4, we have found that the allele responsible for resistance is not present in the introgression lines derived from other background. But when we observe the phenotypic score, it is even less than the GPBD4. The present score for GPBD4 is 4 to 5, and the introgression lines showed a score of around 2, 1, and a few lines showed 3. So from this, we can say that there are some other loci which are responsible for resistance to LLS. If we can tap these new sources, then we can get better resistance than the existing resistance. So we have done phenotyping of the introgression lines, and we have found the resistance. So on further screening, we can confirm them, and we can suggest them into the breeding programs. Thank you.
Thank you, Hima. So we'll have one more presentation, and after that we'll have 10 minutes of question answer for whatever talks we completed. So I request Ms. Surya Murugan from ICRISAT, and she will be talking about the high throughput phenotyping improves selection intensity in groundnut breeding pipeline at ICRISAT.
So, good morning, everyone. So, I'm Surya, Scientific Officer from Groundnut Breeding, ICRISAT. So, today I'm here to present about high throughput phenotyping, improves selection, intensity in groundnut breeding pipeline at ICRISAT. So, groundnut is mainly grown in semi-arid ecologies of Asia and Africa, where the combination of abiotic and biotic stresses. So, optimizing the breeding schema for selection intensity enables breeding programs to realize higher rate of selection genetic gain for targeted traits, as well as to optimize the time and resources. So the selection intensity can be increased by increasing the number of selection candidates. To exercise the selection candidates, we are using high throughput phenotyping and genomic tools. So at ICRISAT, a 2-step selection approach were used, being deployed to improve the selection intensity. LISI scan to select the early vigor plants and manage stress environment for a drought screening. So here comes the methodology. So after the hybridization, the F1s were confirmed with hybridity confirmed with QC markers, and the population of about 104 families were advanced to F2 to F4 using single seed design method. So and from the F4, single plants were selected, and it is raised as F5 progeny rows and harvested as F6 progeny bulks. A total of 1,645 F6 progeny bulks were screened through LISI scan, as well as artificial foliar fungal disease simultaneously. Then about 322 progeny bulks were forwarded to management stress environment testing. So LISI scan, high throughput phenotyping platform, we use this LESI scan to screen the early vigor plants based on the selection criteria of digital biomass, leaf area index, and plant height, as these traits shows good heritability based on the previous studies. And simultaneously, we screen the same lines in the foliar fungal disease resistance, as LLS and rust are the major disease in groundnut. So we screen the population through artificial infector row method, and we narrowed down the population from 1645 to 322 progenies. And these 322 progenies were taken to managed stress environmental studies. So here we raised two plots, water stress condition as well as well water condition. So up to 60 days, we gave normal irrigation to both condition, and after that, we induced mid-season drought, and we stopped the irrigation for water stress condition. We withhold the irrigation, and we impose the stress. TDR probes were installed across the field to measure the soil moisture content, and intermittent stress were applied from the 60 days of days after sowing in the water stress condition. So after the permanent wilting point reached, we relieve the stress, and we calculated the evapotranspiration rate of those stress condition plants. So you can see the graph that at the actual requirements of the ET was estimated till 60 days, and after that the ET was gradually reduced to the water stress condition plants, and when the stress was relieved, again it attains the normal weight. So based on the stress indices, we selected the screened lines. So the coming to the pod reduction, reduction of pod weight or pod yield in the lines, mostly 70%, more than 70% of the lines, that is around 402 lines from 600 lines, showed 70% reduction, 70% pod reduction in the water stress condition during the pod filling stage of the plants. So based on these results, we identified that ICGV 171044, it is one of the parental lines in 12 selected families among the 104 families. It is a potential genotype for the early vigor. So now this 322 lines of these managed stress environmental studies, it is under genotyping of by using mid-density panel of 2500 SNP markers to genotype it, and we will advance this to stage 1 multi-environmental testing. And for the 1645 lines also, we want to genotype it by using a 5K mid-density array for a genotypic selection model, and this will be categorized and used as a training set and testing set for GS model development and cross-validation studies. Thank you.
Thank you, Surya. Now I request even chair and co-chair if you have any question to the completed talks. And from audience also, I would like to have some questions if you have noted down. So one by one, we'll ask.
Till audience is getting organized, can I ask one question to Surya? So Surya, how do you decide a permanent wilting point to release the stress?
By calculating the soil moisture content, we install TDR probes across the field. So 9% soil moisture content, we will calculate the permanent wilting point, and after that we will relieve the stress. 9%. Yes.
Of soil at what depth?
Sorry?
At what depth?
I couldn't understand. After we observed after 15 days of this imposing stress.
Okay. I have one question to Ms. Poonam Bhatt. So, Poonam.
Yes, sir.
So, you have identified some QTLs for the big seed in groundnut. So,
Yes.
in the genomic regions, do you find any major gene that is regulating the seed size in the groundnut?
This QTL that I have mentioned about A05 region, it at 99 to 102 million base pair, there are many genes. Out of those, we narrowed down this TIFI gene, which could be having a potential role for seed size regulation. So we checked its expression and all its homologs expression into the mutant, and we found 4 are downregulated. So that's how we concluded that this is a gene responsible for seed size regulation.
What about this big seed locus? So this is already reported in many crops that it is regulating. So
Yes, sir. It is reported in Glycine Max, and it is reported in, it is confirmed in Arabidopsis. And even in peanut, a lot of other papers have confirmed the region of this that I have mentioned on A05, about 100 million base pairs. The region is known to contribute for seed size regulation.
Okay, thank you.
My question is to Manasa. You talked about biochar application for Sclerotium rholsi, stem rot. What about other diseases? Is it specific to stem
Ma'am,
rot?
I worked on stem rot of groundnut,
Yeah.
and others, they are working on collar rot also.
Means, is it very specific for such kind of diseases or any other
Anything we
application?
can use, ma'am.
What was the reason to like,
Means when the biochar is applied to the soil, it alters the soil's characteristics. It will increase the water retention, porosity, and it will change the pH. By the basic characteristics of biochar is alkaline. If when the biochar, when the soil becomes alkaline condition, the microbial population gets increased. So when the beneficial microorganism get increased, it will suppress the target pathogen. When the microbial population also increases, they will operate its own mechanism, that is competition, antibiosis, lysis, mycoparasitism, and induce systemic resistance. In this way, it will suppress the soil-borne diseases.
what are the microorganisms that are present in biochar?
Ma'am, any beneficial microorganisms like Trichoderma, Bacillus pseudomonas.
How is it possible? Like the production of biochar is like
It's a tedious process.
tedious, and also like heat is involved. Force,
Yeah.
everything will be killed. Then how the beneficial organisms are there in?
No, ma'am. The final end, after the prepared biochar, we'll made into powder. That powdered one, it will consist of nutrients. That one, when applied to the soil, in the soil already existed microorganisms will be there. When applied to that soil, it gets proliferated. In that way, the population of the beneficial organisms will increase.
But in that case, even stem rot, that pathogen also may be growing.
Means there will be competition between the beneficial organism as well as the pathogen. So when the beneficial organism, it operates its own mechanism, it will suppress the pathogen.
Okay. There is a one question from Dr. Sina Roy.
Hello. Yeah, I have question for me, the Ramarajin. So in terms of the biochar, you have for 1%, 3%, and 5%. So I wonder how you decided that percent.
Sir, how we have prepared that concentrations here?
Yeah, for the 1%, 5%.
Under glasshouse condition, 1% means for 1 kg of soil, we'll be adding 10 grams of biochar. Whereas for 3%, for 1 kg of soil, we'll add 30 grams of biochar. And in case of 5% concentration, for 1 kg of soil, we'll add 50 grams of biochar. It gives 1%, 3%, 5% respectively. Whereas in case of field conditions, normally the per hectare of the soil will be the soil amount will be 2.24 into 10 power 6 kg per hectare of the soil. Per hectare, the soil will be present. So for for each plot, we'll calculate how much amount of soil will be present. And for each plot, we'll calculate 1%, 3%, and 5%. In that way, we have done.
So actually, my question, why you decide the 5%, like not go to 10%? Why you decide is 5%, not go to that 10%?
because why 5% we have decided till 5% you are asking, sir. Yeah. Means if we go more than 5%, the production we need, it will be tedious. The amount is very less, so we have stick on till 5% only. Means 3%, 5%. We need more amount of biochar. For preparation of biochar, it is a very little bit tedious process. It almost took for us to prepare, it almost took for 2 months for preparation of this biochar. So we have stick till 5%. We can go more than 7%, 10%. But if we increase the concentration of biochar, the soil properties also, it will be changed. It will be in negative, there will be negative effect also we can able to see.
Okay, we'll take one more question from Dr. Sanjuti Sinoroy. So Charles, behind you, there is you.
I have a comment for Mansa. So this is a novel studies and what you are proposing, it's your hypothesis, right? The microorganism is changing. So you need to have microbiome data. You need to have like, you should state it like it is an hypothesis. This is not a proof.
No, ma'am, we have done qPCR, means we have isolated soil DNA, and we have checked the actinomyces and bacterial population was present. But we didn't go for metagenomic study.
Yeah, you need a quantified data.
Yeah,
right?
metagenomic study we didn't go. So we are doing that one also in future. So
Okay, thank you.
Till that we have sticked.
I have a question for Rati. So when actually you were mentioning that you have a population which is having VP porous domination and have a dormant population. So this is for my understanding. So yes, VP porous domination is a problem, right? But how much dormancy factor should be ideally present in a groundnut population that will be helpful for the farmer?
Ma'am, 2 to 3 weeks is desirable. So the reason for 2 to 3 weeks being desirable is because maximum farmers will resow their harvest. So if the duration is more, like one of the parent TG22, the dormancy period is 45 days. So it is not desirable. Plus it is a late variety also. So the objective was to look for a dormancy period for 2 to 3 weeks plus have earliness, which is from the parent TG51. So I was looking for goodness from both the parents. So that is.
Thank you.
Thank you.
Thank you, Rati, and thank you all for all the questions. We'll move to the next round of presentations. And now I would like to call upon the stage Khaja Moinuddin from UAS Raichur, and he will be talking about the candidate gene discovery and marker development for fresh seed dormancy in the groundnut.
Good afternoon to everyone. I am Khaja Moinuddin from University of Agricultural Science Raichur. I am here to present one of my research objectives, that is candidate gene discovery and marker development for fresh seed dormancy in groundnut. Coming to the introduction, as you all know that groundnut is a segmental allotetraploid having a chromosome number 40, and it is native to South America, popularly known as the king of oilseed. Groundnut has highly evolved for its nutritional and economic importance. Despite of numerous benefits, groundnut cultivation faces challenges with pre-harvest sprouting, which is germinated before harvesting only at maturity stage. In order to get rid from this, we are going to identify the genotypes which is having the dormancy period of 2 to 3 weeks. So for this we used genome-wide association studies. So where we did the selected the population, that is 184 mini-core lines from ICRISAT, and we did the genotyping. The trait is days to 50% germination, and next we move on to the genotyping. We did genotyping for 184 mini-core lines, that is whole genome resequencing, and did the statistical analysis, that is GWAS, and we conducted the validation part also. So these are the Manhattan plots which are showing the significant MTS for fresh seed dormancy. Here we can clearly see that the chromosome number 5 showing significant MTA in all the models in all the seasons. So we did genotyping for 4 seasons at 2 locations. So we taken 2 rainy seasons and 2 post-rainy seasons. These are the SNPs and its position and annotation for gene ID. So chromosome number 5 and 15 and 1 and 4 are showing significantly related to the dormancy trait. So this is the pathway. The ABA, abscisic acid, is directly related to dormancy and gibberellic acid is related to germination. So when we are having the more ABA in seeds, so it will leads to dormancy. When we are having the more amount of gibberellic acid, it will leads to germination. So here which are highlighted in red color. So those genes are related to dormancy and which are highlighted in green color. So those genes are related to germination. So after that, we did the allele mining for entire sets. So which are showing favorable allele calls, we selected and we sent for validation. Among them, we got 8 which are showing clearly differentiation between favorable allele and unfavorable allele. Among them, we developed 8 CARSP markers. So these are clearly showing the differentiation between dormant and non-dormant. So these developed and validated markers which can be used in the marker assisted selection. Thank you.
Thank you, Khaja. Now I request Sri Raksha to come on the stage and she will be talking about the molecular dissection of heat tolerance through bulk segregant analysis approach in groundnut.
Good afternoon, everyone. Today, I am presenting my research study on molecular dissection of heat tolerance through bulk segregant RNA sequencing approach. In the present climate change scenario, it is very important to know the molecular mechanism behind the heat tolerance so that we can go for the development of the varieties which is resilient to the heat tolerance. So in this context, we have employed a BSR-seq approach to identify the candidate genes which are responsible for the heat tolerance. Here in this study, we have taken a panel of heat tolerant and sensitive genotypes, which are selected from the real mapping population, which were derived from JL24, which is heat sensitive parent, and 55437, which is heat tolerant parent. Then to simulate the heat stress condition, we have maintained the selected genotypes in environmental growth chamber. So where the temperature was raised up to 45 degrees Celsius, then the RNA was isolated from flower and pod tissues of those selected genotypes and pooled as a heat tolerant bulk and heat sensitive bulk and subjected for the BSR sequencing. And then the sequencing reads were further processed using bioinformatic approaches like I have used CLC Genomic Workbench as a platform, and further validation was validation of the candidate genes were done through QRT-PCR. As a result, totally 155 DEGs were found in flower tissues and 1097 total number of DEGs were found in pod tissues. And some of the very important transcription factors were identified, which were having the role in stress adaptation. And further, the gene ontology enrichment analysis revealed that iron-ion binding genes are mainly enriched, functionally enriched in flower tissue, and hydrolase activity genes are mainly functionally enriched in pod tissues. The main, the key finding of this study is identification of the important gene that is phosphatidate transferase, which is having the role in minor minor phospholipid biosynthesis, and it is having the stress response, uh, uh, having the role in stress response, and which showed very high fold change difference, that is 815 fold difference between the heat tolerant and sensitive genotype. So it is showing that it is very important gene for the heat stress mechanism. So we went for to better understand the functional relevance of this gene. We went for protein-protein interaction network analysis. This gene revealed that it is interacting with other genes and having the role in lipid membrane lipid remodeling mechanism. So it is very important gene for this is very important gene while the heat stress is employed. So some of the studies which is having the relevance to our study, like phosphatidate citrial transfer is having a very important role in stress responses and as well as signaling pathways. And further, QRT-PCR validation was done between the tolerant and sensitive genotypes. This gene showed a very high expression when comparing to the sensitive genotype. And then we went for SMP analysis within the DEGs. So very good number of polymorphic markers are we found within the DEGs. And these can be a potential markers for further studies. And then we went for comparing the transcriptome studies with already published data from genetic mapping. So this is also showing the same as the results. Some of the genes are overlapping with the transcriptome study. So this is the final outcome of our study, that PCT gene can be, we can, after the validation, we can go for the marker assisted selection. Thank you.
Thank you, Sri Raksha. Now I request Mr. G. Nehru, and he will be talking about the marker-assisted selection, quality control, and METs in groundnut breeding pipeline at ICRISAT to enhance the genetic gain.
Greetings all. Myself Nehru, PhD scholar from ANGRU, and presently working at ICRISAT groundnut breeding department. My topic is about marker assessor selection, quality control, and METs in groundnut breeding pipeline at ICRISAT to enhance genetic gain. How this SNP markers and the marker assessor selection forward breeding works. With the help of this, how the groundnut breeding pipeline working at ICRISAT, I am going to explain in this presentation. And we use low density assay markers with the Intertech platform and high density markers with the thermofisher platform. And these markers are used for parent selection, hybridity confirmation, forward breeding, line purity, and environment characterization guided by multi-environmental strategies. The purpose of these all markers is to increase the rate of genetic gain in groundnut breeding program at ICRISAT. And how our programs work. First, we select the parents with the desirable traits. The desirable traits may be high oleic, biotic and abiotic resistance, drought tolerance, or heat stress tolerance. All this. The parents were the two parents which are different desirable traits we have crossed after getting F1. Firstly, we have to know that the F1 is true F1 or self-food plan. For knowing the true F1s, we go for a 10-SNP panel for hybridity confirmation. We can see in the slide, the two that with help of 10-SNP panels, parent 1 and 2 were homozygous. In F1s, we have got heterozygous allele. The F1 which has a heterozygous allele is a true F1. After confirming the true F1, we go for forwarding to F2 to F5. For each advancement from F2 to F5, we go marker assessor selection with QC markers and 49 trait specific markers. After that, the F5 is forward to F6. From there, it is called as advanced breeding lines. And these advanced breeding lines were sent to METs through various locations in India. And ICRISAT sends its MET at Telangana, Gujarat, Rajasthan, Karnataka, Odisha, and Andhra Pradesh. After METs, we go for mid-density genotyping for genomic selections. And after that, we will identify the superior lines. Again, these superior lines can be used as parents for different line development. And from that, we have presented here one MET conducted at 5 different locations with 2 HPUs, homogeneous production unit. And we have 5 locations: Bikaner, Targadia, Junagadh, Palem, and Patancheru. This Bikaner, Targadia, and Junagadh comes under HPU2. Palem and Patancheru comes under HPU6. And we have gone to analysis with multi-state stability index. Out of that, one is a Smithel-Hazel index. From these methods gives that the traits, the genotypes which is specific to that HPU and common genotypes for both the HPUs. In these slides, we can see that HPU2 has specific genotypes identified: G10, G13, G16, G21, and G29. And HPU6 has got best and stable genotypes: G3, G7, G13, G16, G25, and G29. And the common genotypes for both the HPUs are G3, G13, G16, and G29. And the genetic correlation was also, there is a high genetic correlation between pod yield and seed yield in both HPUs. And the selection gain percentage also high for both pod yield and seed yield in both HPUs. In this way, we go for multi-trait selections and we identify stable genotypes. And the way forward, the SNP markers can be used for hybrid confirmation, mid-density genotyping for selection of desirable traits at different population and METs. We can ensure the target breeding ensures the yield, sustainability, and trait stability. The multi-trait selection can be used for gain genetic gain. Thank you.
Thank you, Nehru. Now I would like to call upon stage Ms. Priya Upadhyay from NIPGR India, and she will be talking about exploring the evolution of symbiotic hemoglobin in peanut, a narrative on the evolution of plant globins.
Hello everyone. Myself Priya, a PhD student at NIPGR. Welcome to the innovative journey of how the evolution happened in peanut when we talk about globins. So as you can see here, we all somewhere heard about symbiotic interactions. So plant bacteria interact. In turn, this nodule formation happens. So inside this nodule, the nitrogen fixation happens with the help of this bacterial enzyme, which is nitrogenase. But this nitrogenase is an oxygen labile enzyme. So plant have to maintain a minimum oxygen concentration inside this nodule so that this nitrogenase can work efficiently. That is why plant express leg hemoglobin inside the plant nodule, similar to our blood, as we have hemoglobin, as it transport oxygen to our blood. So this leg hemoglobin sends the oxygen, it binds to the oxygen and maintain a minimum oxygen here inside the nodule so that this nitrogenase, which is a nitrogen fixation machinery, it can work efficiently. But this leg hemoglobin has to maintain a specific structure so that this oxygen can bind easily and dissociate easily because it has to maintain a minimum oxygen inside the nodule. That is why this leg hemoglobin is always in a penta-coordinated structure so that it can transport oxygen to the bacteria so that they can respire, and also it can maintain a minimum oxygen inside the nodule so that this nitrogenase can work. So what about peanut globin? Is it the same hexacoordinate or pentacoordinate structure? So in the RNA-seq, what we have found that there is no leg hemoglobin genes in peanut. Further, when we did this phylogenetic analysis, we found there are 5 globin genes in peanut which cluster with class 1 non-symbiotic globin. So here a parallel evolution has been happened, like this leg hemoglobin has been evolved from dimeric hexacoordinated structure to a monomeric pentacoordinated structure. But in peanut, what we have found, this hemoglobin has been evolved from class 1 non-symbiotic globin, which is a dimeric structure as well as hexacoordinated structure. So as it is nodule specific, so we have designated it a name, that is symbiotic hemoglobin in peanut. So what we have found in peanut, I purified this protein symbiotic hemoglobin and found a characteristic sorted spectra which confirms the heme group present in peanut globin. Further, I have found on reducing this protein, it is showing a characteristic Q spectra which confers that it is a hexacoordinate globin. In the side exclusion chromatography, I have found this peanut symbiotic hemoglobin came out to be a monomeric protein and hexacoordinated structure. So the question arises, is it is behaving the same as leg hemoglobin? So we have got these seeds, Lotus japonicus CRISPR lines. They have deleted all leg hemoglobin genes. I have complemented these lines with Lotus leg hemoglobin itself and peanut symbiotic hemoglobin itself. And I found that yes, peanut symbiotic hemoglobin is doing the same function as the leg hemoglobin. Further, I have to validate this experiment by doing kinetics experiment as well to show that whether the oxygen binding and dissociation affinity is same or not. Further, in the synteny analysis, I found that peanut symbiotic hemoglobin have evolved from this non-symbiotic globin 1. So I have purified this protein, did the spectra analysis and found that this non-symbiotic hemoglobin, when I have oxidized, its conformation changes fully. And also it came out to be a dimeric protein. So the question arises, how this evolution has been happened from the dimeric non-symbiotic globin genes to the monomeric symbiotic hemoglobin genes. So my future plans are further to study how this evolution has been happened by mutating the conserved dimeric interfaces of this non-symbiotic globin so as to trace the evolutionary pathway of globin in peanut. At last, I would like to thank my supervisor, Dr. Sinjyoti Sinha Roy, for all the guidance, my organization, NIPGR, UGC for the funding, and my DBT Institute for all those funding related things. Thank you.
Thank you, Priya. Very good energy and well in time. So I request Dr. Madhavi Sharma, and she will be talking about identification of genomic regions and candidate genes for high resveratrol content in groundnut.
Good afternoon, everyone. I'm Dr. Madhavi Sharma, a DST scientist at ICRISAT. So I'm working on identification of genomic regions and candidate genes for high resveratrol content in groundnut. So resveratrol, being a phytoalexin, it has so many health benefits. It acts as an antioxidant, anti-inflammatory, anti-carcinogenic. And groundnut, it is a very good source of resveratrol. So you can see the role of resveratrol in biotic and abiotic stresses. So this is the graphical representation of it. So we have taken 184 mini core collection and we did the resveratrol estimation in those 184 mini core. So out of these 184 mini core, these are the genotypes having high resveratrol content ranges from 14 to 3031 μg per kg. So then we did the GWAS analysis by both 58K array and WGRS. And the highlighted one are the key candidate genes that are found on chromosome 2 and 12. So this is the GWAS analysis by 58K array. And this is the GWAS analysis by WGRS data. So the candidate genes that we got, out of which few were having the important role in biosynthetic pathway of resveratrol that is already existing. So this is the biosynthetic pathway. So the highlighted one, yeah. So as you can see, this transcription factor EFE1 that was found on chromosome 2 here. So what it does, it converted coumaryl CoA into resveratrol. And this zinc iron binding protein that we got on chromosome 2nd, so what it does when there is any stress, so there is an enzyme still in synthase. What it does, it converts the, or it synthesizes the resveratrol. So, and this zinc iron binding protein, it acts as a cofactor that plays a structural role in stilbene synthase. The second one is beta glucosidase. So it is the key enzyme that converts the polydatin, that is the same, it is the similar phytoalexin that has almost similar structural similarities with the resveratrol. So what it does the glucosidase hydrolysis of polydatin and remove one glucose moiety and convert into resveratrol when there is a stress. So let's move when there is a stress. So I will highlight on some of the points of resveratrol, why it is important. As I said, it has so many health benefits. Health benefit most important is the antioxidant. And you know, antioxidant, it plays a great role in human health. And it is having so much of importance in food industry. So that's why we have targeted this resveratrol content. And moreover, resveratrol, it is present in very few crops, mostly 3 to 4 crops, groundnut, berries, grapes. So, also this resveratrol, it is synthesized in very less quantity. So our main aim, why we have targeted the this particular trait, because it is synthesized in very less quantity in resveratrol, in plants. So by targeting those genes that we identified, we can upregulate those genes and upregulate those genes and can synthesize it in more quantity, so that we can suggest it to the food industry. And moreover, in recent studies, it has found that resveratrol, it plays a great role in cardiovascular and neurodegenerative diseases like Parkinson's disease and Alzheimer diseases. So yeah, and there is not that much work on resveratrol when it comes to the finding of genomic regions. So yeah, we try to do, and hopefully we will find out the key major genes that we already found, and the markers that are responsible present on chromosome 2nd and 12 will go for the validation of it. And when we'll get, after validation, we will go for the haplotype analysis. And when we'll get the authenticated data, so we can suggest it to the food industry, and we can use it for the marker assisted selection. So this is our overall target. So this is my team. So thanks to genomics team for all their cooperation and support. Thank you.
Thank you, Madhvi. So we have Dr. Anant Kurela, and he will be talking about the distribution and risk assessment of Aspergillus flavus infection in major groundnut growing regions of India.
Good afternoon, all. I am Anant Kurela, PhD scholar from the Groundnut Pathology Lab at ICRISAT. The next 5 minutes, I will be giving a talk on this topic, that is distribution and risk assessment of Aspergillus flavus infection in major groundnut growing regions of India. To give a brief introduction, yesterday a few delegates have raised this concern, like aflatoxin, its impacting the export trend, especially we are not able to export to European Union because of the strict guidelines of the aflatoxin. So almost with the same background, my PhD work is going on to map the high-risk areas of this aflatoxin production in major groundnut growing regions of India. So a little bit introduction. The Indian average yield is around 2075 kgs per hectare, which is good when compared to the global average, like which is at 1648 kg per hectare. So then what is the problem? Even though we are producing the groundnut in good quantity, but as already mentioned, we are able to export only around 8 lakh tons every year. So we have a potential to export more quantity, but still, what is hindering? One of the major concern is the aflatoxin production by the Aspergillus species. So we know the problem. So what will be the solution? So first of all, there is a need to map high risk areas, so that once we identify these high risk areas, which is means where the aflatoxin production is more, then it will be helpful to devise management strategies that are region specific, because India has diverse agroecological zones. So under this study, we started to collect the pod samples from almost 11 states, which are we identified as major groundnut growing region states in India. And we collected around 229 pod samples covering 36 districts in 11 major groundnut states of India. So once we collected all these pod samples, we started with kernel infection percentage. So we have took the mean kernel infection percentage from each state. So here we can see Gujarat and Punjab and also Maharashtra showing the highest kernel infection percentage compared to other major groundnut growing regions of India. Also, we try to classify all the districts, that is 36 districts. We try to group them onto like safe, acceptable level, and also moderately toxic and toxigenic range of this kernel infection percentage. So almost these districts are around 10 to 15 districts are in safe level, where the kernel infection percent is from 0 to 15%. And then these districts are in the acceptable levels of the kernel infection percentage, which is in the range of 15 to 30%. And the last category is moderately toxic, which is around 30 to 45% of the kernel infection. Here, only 3 districts, and Jalgaon is showing high kernel infection percentage compared to other districts. And we have also one more category that is highly toxic, but surprisingly, from all the 11 states, 36 districts, we didn't get any districts showing more than 45% of the kernel infection. So the conclusion would be this is a preliminary study. We are just seeing the presence of the Aspergillus flavus from these 229 pod samples collected from overall major groundnut producing states. So the conclusion is like, see, this kernel infection percentage is showing in is very much varying from state to state because of several factors like farming practices, soil microbiome, and host susceptibility. So once we identify these high risk areas with if we can quantify this kernel infection percentage with the ELISA, indirect ELISA, where we get exact aflatoxin levels, this will really provide a solid or concrete data to map the high risk areas in the major groundnut producing states of India. Thank you.
Thank you, Anant. Now we have last flash talk of Mr. Nikhil, and he will be talking about identification of candidate genes and markers for kernel iron, zinc, and protein content in groundnut.
Good afternoon, everyone. Myself, Nikhil Sagar from Angro, India, and I am going to present my work of identification of candidate genes and markers for kernel iron and protein content in groundnut. And groundnut is popularly known as the king of oil seeds. It is a self-pollinated segmental allotetraploid, and it is native of Brazil, South America. Groundnut is rich source of several mineral nutrients, essential amino acids, vitamins, and it has significant amount of bioactive phenols, flavonoids, and isoflavones. And consumption of even small quantities of groundnut can provide more energy like 567 kilocalories as per USDA nutrient database. And the key nutrient mineral contents in groundnut includes iron, zinc, and protein content. And consumption of 100 grams of peanut can provide iron of about 4.58 mg and zinc 3.27 mg and protein content of 26 grams. And iron helps in oxygen transport and it also regulates cell growth and development. Whereas zinc helps in the synthesis of RNA and DNA and it acts as a vital cofactor for numerous cellular processes. Whereas the protein content, it helps in emulsifying activity and helps in the water storage capacity. And the major prevailing problem, particularly in the African countries, includes malnutrition or the hidden hunger. And consumption of groundnut helps to overcome this problem. And the major cause of malnutrition is the improper intake of vitamins, minerals, and the consumption of groundnut can overcome this problem. And it has high protein content than any other true nuts. And I have carried out the GWAS analysis using the phenotypic data of five seasons across 2 different locations and the whole genome resequencing data and carried out the GWAS analysis. And the phenotypic variability analysis also showed that there is a large variation in the iron and zinc content across different seasons. And the GWAS analysis showed a total of 59 significant MTAs, where I have mentioned only the key significant MTAs harboring the key candidate genes. And for iron, there are the MTAs that are harboring key candidate genes like NAC domain protein, zinc or ring finger protein, whereas for zinc, MYB transcription factor and ring finger protein helps in the iron and zinc homeostasis pathway. And the key candidate genes associated with the protein content includes the aspartic proteinase and protein serine threonine kinase. And these candidate genes helps in the iron and zinc homeostasis pathway, where these genes helps in the absorption of more contents of iron and zinc from the soil into the root cells. And from the root cells, these iron and zinc are activated and their uptake is enhanced by key components like NAC domain or ring finger proteins, as well as the MYB transcription factors. At last, these iron zinc homeostasis pathways helps in the more accumulation of the iron and zinc in the seed kernel, where it is stored in the endosperm. And from the MTAs I have got from results, I have selected 9 MTAs based on the SNP call variations. Of these 9 MTAs, I have developed markers and sent, and I have validated these markers. And the 3 markers were successfully validated among the 9 Cas markers developed. Of these, 2 include the markers for iron content and 1 includes the marker for zinc content. And these MTAs were harboring key candidate genes of about 90 candidate genes. And the key candidate genes in the iron zinc homeostasis pathway include the NAC domain protein, MYB transcription factor, and ring zinc finger protein. Among the 9 Cas markers developed, these 3 markers, 2 for iron and 1 for zinc, can be used for further marker assisted breeding. And from the candidate genes, we can obtain the haplotypes, which could further be used in the haplotype based breeding for developing nutrient rich groundnut varieties. Thank you.
Thank you, Nikhil. We'll have quick 3 questions. So from audience, yes, Dr. Ajay PC would like to ask a question.
Yeah, one suggestion to Anand Kurela. I think for his aspergillus flavor sampling, he has collected samples from different parts of India. I think it is better to, if he can collect samples from Rajasthan, especially the Bikaner region and then Mando region, and also the Bundelkhand region between UP and Madhya Pradesh, because they are the almost they are the second and third position with respect to area.
Yes, sir. Actually, the sampling is still in pipeline. We are already received samples from Rajasthan, and also we are having a plan to collect samples from Madhya.
Thank you, sir. From chair and co-chair, if you'd like to
I
ask.
have a follow-up question to Ananta. you are classifying based on the percentage of infection, but are you looking at the level of infection?
We are going for a plating of around 30 kernels. So out of the 30 kernels, we are see how many seeds are getting that in means by, and that we are converting into.
For Ananta. Actually, you are looking at different regions, but are you also looking for which genotypes they are growing? Because it may be different with the and just based on the seed infection, we cannot tell that. Many a times, aflatoxin production may be different as compared to the
Yes, yes, madam.
infection.
That's why in the last slide I was quoting the same. But with this kernel infection percentage, we cannot say whether it is really a toxigenic or atoxigenic. So again, we need to, for assays like ELISA, know the exact aflatoxin production of this particular isolate.
And also what seasons you are targeting?
What?
Season.
Season, we are targeting both rainy and post-rainy. Because in we take the example of Telangana, it is a post-rainy. Andhra Pradesh, it is the rainy season. And in Gujarat also, majorly grown.
But you are analyzing differently, right? Or you are pooling all the data?
Right now, we are pooling.
It's no, we need to analyze separately, I think.
Once we get the ELISA data, we definitely we will.
Thank you, Ananta. So we are already 15 minutes late. So what we can do in lunchtime, we can, or sir, cases Naik, last question we'll take from Dr. Cases Naik.
This is to Madhavi Sharma with regard to this resveratrol content. So, are there any genotypic differences in the resveratrol content? And my second question is there any relationship between resveratrol content and drought resistance? Stress resistance.
Yeah, I'll answer your second question. Yeah, resveratrol, it has a very important role in that antifungal infection. So this A flavors, it is having negative correlation with the aflatoxin. And sorry, your first question?
Genotypic differences among the genotypes. Are there the
Yeah, there was difference. There was a range of around 16 to 1600. So yeah, there was variation in the genotypic. At genotypic level, the concentration varies. Yeah. Thank you.
Thank you, sir. So rest of the question you can ask during lunch time break or maybe in tea break. So I'm thankful to both chair and co-chair of the session. Thank you to all young researchers for impressive work. I wish you good luck and congratulations again. Thank you.
Ladies and gentlemen, here we complete the session on next generation researchers. Looks like our next generation is ready for the world. Amazing confidence and the depth they carry on their subject. The proceeding forward, we'll go with the award ceremony and then break for lunch. So yesterday's poster display, around 26 posters were displayed and 5 best posters are shortlisted. Amazing work done by Dr. Naveen Puppala, Dr. Ran Hawau, and Dr. Ajay BC in shortlisting the best of 5 posters. Any guesses who have got the chance or the spot for the award? Any guesses from the audience? All right. I now request Dr. Stanford Blade, Deputy Director General, Research, ICRISAT, to give the awards. Also invite the Chair AAGB 2025, Dr. Manish Pandey, to accompany him on the dais, please. Ladies and gentlemen, and the award goes to Ms. KL Vidyashri, Groundnut Pathology, ICRISAT. Miss Poonam Bad, Bark, Mumbai. Dr. Vinay Sharma, Groundnut Genomics, ICRISAT. Miss Priya Upadhyaya, NIPGR, New Delhi. And Miss Muktambika Kuruva, Groundnut Breeding, ICRISAT. Can we have all the poster presentation winners on dais, please? Appreciating all the poster presenters for presenting and the winners for taking their spot. Moving forward, around 13 flash talks were accommodated in the program, and the 5 best are being shortlisted. Meanwhile, I take pleasure in thanking the evaluation team, Dr. Naveen Pupala again, Dr. Ran Hawav, and Dr. Kenta Shirashava for evaluating the flash talks. Thank you all the presenters for presenting well, taking your time out and coming here and presenting. Meanwhile, I get the list. Let's keep the surprise alive. Any guesses from the audience who will be winning? Not the poster presenters again.
Thank you very much for the excellent work. I just want to acknowledge as well the superb activities of our next generation researchers. I would share the sentiment of just how positive I am about the next generation of work when it comes to all things groundnut research and beyond. It's a great pleasure to acknowledge all of the students, and I'll spend a little time with them, I hope, afterwards as well. The very fact someone is running in with the information probably means we have some certificates from the talks that were given this morning as well. So I just want to thank both the poster presenters and of course people that were giving oral participation this morning with their presentation, just how grateful I am. And I think a round of applause again would be very appropriate for all of our presenters. I would just observe also, I hope that there's no wagering going on in the crowd or there's no lottery on who's going to win at this time. But I'm very much looking forward to the 5 very successful people that made presentations. But to be clear, everyone did a superb job. So thank you.
And the most awaited moment, we got the results of the flash talks, and the winners are Miss Rati Satish. Mr. Kaja Moinuddin. Miss Madhvi Sharma. Miss Surya Murugesan. And Miss Ramaraju Mansa. Requesting the winners of the flash talks joining on the dais, please. Ladies and gentlemen, whether you have noticed or not, the ratio of girls is to boys is 4:1 in both the poster as well as flash talks. And a big round of applause for the girl power, also for the boy power for competing equally. Thank you, one and all. Thank you, Dr. Stanford Blade. Thank you, Dr. Stanford Blade, for giving away the awards, and thanking Dr. Manish Pandey for accompanying here. We'll break for lunch now. After lunch, we will begin our session around 2 and requesting one and all to be present in the venue by 2. Requesting all the students to stay back here. Ladies and gentlemen, welcome back for the post-lunch session. Before we move into session, I request our organizing team to play the video of Bombay Super Hybrid Seeds Limited. Can we have the video, please?
Yeah, hello. The purpose here to show there is one aspect is industry R&D, other is extension, and third is industry and farmer. So how actually this groundnut industry is spreading, the kind of machineries requirement, kind of the farmer preference that are briefed here in my small video, because this journey, how the R&D we do, a lot of things are there, but how actually it should reach to the farmer level. That small concept of processing and everything, I was trying to cover it for the education of the people, because ultimately the end user farmer requirement and how the things are moving, that should be clear in the mind. Okay, thank you, sir.
Thank you, Dr. Hemang Bakshi and team. It was a wonderful video bringing in the industry lens of groundnut R&D and the scale we operate. It's always pleasure to see, and I'm proud to say that every iota of your hard work be it a younger researcher or the experienced researcher, whatever contribution you do here, we're targeting a very large impact that is global food security, and we should be congratulating ourselves for being a part of this community and contributing so much. Moving forward, we are opening the session on genomics assisted breeding. I request Dr. Navin Puppala to come to the dais. He will be the co-chair of the session, who will be shortly joined by Dr. P. Janila, the chair. Over to you, co-chair.
Thank you. Good afternoon, everybody. I hope you had a good lunch, and I hope you don't fall asleep. So we'll start our session with genomics assisted breeding. The first talk of this afternoon is genomic tools for groundnut genetic improvement. This will be presented by Vinay Sharma from ICRISAT.
So, very good afternoon to everyone who was present here, and good morning and good evening to the delegates which are joined from all over the world. So, today I'll be presenting what are the genomics tools and the groundnut technologies which are helping for the groundnut genetic improvement. I won't bore with you the journal usual introduction of the groundnut. You are hearing it from last 1.5 days. So we all know it's healthier and nutritious, and it can work out for our dietary, and it's also we use it as a fluffy. So today I am here to make a dive with our available tools and technologies available in groundnut genomics to accelerate the groundnut breeding. So I'll just give you the just the introduction how we are moving it forward from the oilseeds to the most delicious and the nutrition rich crop, then oilseed crop. And here we also know these are some example I put here to be how we can use the groundnut nowadays, except including the oilseed crop. So this is just an overview I put without any usual introductions. So one of the biggest challenge, the looming perfect storm. So in this, the climate change, land degradation, loss of biodiversity, food crisis, energy crisis, and the population explosion. So this is just a complete package, and it just impact our crop. And how we have to deal with it. So for that, we have made this genomics and just try to be used as application for the improvement and accelerating the groundnut breeding. So this slide I have put from the morning itself is showing how the climate change impact and affected the whole world, and most probably the developing countries are most affected from the climate change than the developed countries. And most interestingly, that entire groundnut production area globally comes under the adverse climate change is impacted highly, and it make the yield penalty between percent and up to 25. It comes around but decreasing to -20. Yeah. So here I just give the overview of how we can just move forward with the available genetic and genomic resources, what's the need, how we have what the prerequisite for the genomics, and how we can use that resources and just go further with that. So here I'm just moving with these two to improve the genetic enhancement, how we can go and maximize the genetic diversity, because it's a prerequisite requirement for developing the genetics and genomic resources. So we had to improve the genetic enhancement. We need germplasm and the genetic variation available for that particular trait. If we don't have a diverse germplasm or the genetic variation for the trait, it's very difficult to develop the genetic the genomic resources. So here, what strategies we are using? We just perform the large number of processes, just try to make the large segregating breeding populations, early generation screenings, genome-wide predictions, mass MABC, rapid generation, and the genome editing. And for maximizing the genetic diversity, we had perform the whole genome resequencing of the germplasm collections and phenotyping using high throughput platforms and simulation and crop models, which we can use to improve for the biotic and abiotic stress traits and the development of the diagnostic marker, which is directly linked with the gene of a trait, a particular gene of a trait, and the gene discovery. So this is how we can just use our available genetic resources in the translational form in the crop. So here we are just using the global germplasm collections. We had in ICRISAT, we had available reference sets, mini core collection from the core collections. We had the genetic population, real nils, everything, elite cultivars, mutant population, precise phenotyping and genotyping data, and genomic resources. We had now available the reference genome for both diploid and tetraploid. Core and pan genome is also recently developed, and the resequencing of germplasm we had recently completed for 2,000 lines. In coming slide, you will get the information how much candidate genes, candidate gene discovery, and diagnostic marker development for the different traits. And we also developed the cost-effective, high, mid, and the low-density genotyping platforms, which can help you to accelerate the breeding. And these are, I just put what are the applications. We can just get it once these resources are available, what informations we are getting from that. So what are the genomic application we can do with that? So it can help you out to just make the duplication, what is available in the gene bank. We can just make the purity and all those things with the help of these markers, available markers, candidate gene discovery for the genome editing, marker assist selection, which is routinely used in the breeding program, and the genomic selection for the prediction and the identification of the superior genotypes, which we routinely used in crosses, and the elimination of the linkage drag and the crosses, and the hybrid conformation, which is routinely used for selection of the individuals for the selection, and the checking the seed purity in the seed system. So that is thing. So today I will just cover these three topics. What are the genomic resources available in groundnut, and what are the genotyping platforms available with us, and what are the gene discovery and the diagnostic markers available for different traits. So first of all, I will just give you what are the genetic and the genome assemblies available in our last 1 decade from diploid to tetraploid species. So it started in 2016 when we have, with the help of the partners from the USDA and the Chinese, we had sequenced the diploid progenitor genomes in 2016. And after that, we move forward with that. And in 2019, we have the availability of the tetraploid genome of groundnut available and in the two different species, in Tifrunner and in the Shitoki. And nowadays, right now, ICRISAT collated around collated 4 reference genome out of as a main lead and 3 as a collate from the 8 genome sequencing. And these high quality genomes are global resources and being used by all the genomics and breeding researchers across the globe. And now we had the efforts moving forward with the EIB, and we are just making a new reference genome with the ICGB IS1388 in the Spanish bunch and 01276 together with the EIB platform. So maybe these 2 new genomes are available in the coming days. So we have a bigger things too, how we can just utilize, as Dr. Kuldeep Singh yesterday mentioned, we don't have that much the description. We are in the preliminary stage for the information for all the core collections available at. So we know how we can harnessing the novel things and the resources from, and just track the favorable alleles for that to be to get the things bring it into our to the cultivator breeding program. So for that, recently we had completed the sequencing for ICRISAT groundnut reference set. So in that we had sequenced 264 cultivated and 36 wild accession at 10x, and we got around 25.38 million SNPs, and in which the 1.1 millions are in else. And the other 2,704 gene-based presents abscess variations also. We also developed the pan-genome from it and are getting around 82.42 MB additional sequence identified from the available reference genome. So now it's resulted around 2.5 around 2.5 GB, which resulted which resulted in a cultivator pan genome of now is around 2.6 GB. And this effort is ongoing, and we had done with the preliminary analysis, and we had performed the whole genome sequencing of diverse lines of core collections of 1,500, and the preliminary analysis and the whole genome resequencing of this one is completed. We are getting around around approximately 9 lakh SNPs, 9 lakh SNPs, which we use for the further analysis of the discovery in our breeding program. We also developed the gene atlas, which is provide the baseline for the system biology kind of stuff to be utilized in the functional genomics. So this is what are the genomic resources available. So here I will just provide what are the cost-effective platforms available with us at different platforms. So we had these are the 4 genotyping platforms available at the 3 different platforms: 58K SNP array, mid-density 5K array, and 2.5K array, and low-density 20 SNP, which is routinely used in our breeding program. So this high-density 58K SNP axiom array is developed in 2017 using the whole genome resequencing data of 30 cultivated and the 11 wild deployed progenitors. And we do the filtering and come across after that we are getting around 58,000 SNPs, which are routinely used in the application of DNA fingerprinting, diversity analysis, QTL mapping, and we also deployed this one in the genomic selection also. So this is the platform we routinely use. But the problem is with this platform is that it is based on the deployed genome. So with the availability of the tetraploid genome sequencing in 2019, we had just select the markers from this 58K. So what are the markers which are showing high PIC? We just select the marker from there and just align that marker with the tetraploid assembly. And after that, we just design this panel. So this is the background for that 50K. It's really a very little bit high cost, not a cost effective, and it's not be that too high for the routine breeding program. And the application like background breeding, genomic selection, not need to be high density genotyping assay. So we had to be developed some economical, which even we used in our routine breeding program. So this assay is developed on that one, and we had collect a marker 5,000 markers from that one, and included the 81 trait specific markers coming from the different studies of QTL-seq and the QTL-seq mapping GWAS studies. We included those markers also in this panel to be track the favorable alleles. So good thing is with that, the cost for the 58K assay is around $50 to $60 per sample. So we had decreased that directly to the cost of $10. So with this one is now routinely used by the groundnut breeding community across the world. And this is the freshly cooked article in the Plant Genome Kitchen, and maybe available in next 2 days with online. So you'll get the information later to this article, how it is developed, what is the basic behind that. Everything is available in this article. We also deployed this assay with the applications. We successfully implement in the mid-density assay in the genetic diversity analysis. We use different sets of genotypes, reference set, founder parents, which are routinely used in our breeding program, and core collections, and this one, and we deployed, and we will get the clear clusters based on our these sets, diverse sets. We also utilized it in the background genome recovery using mid-density assay. So here we will perform the genotyping of MABC lines using 58K and the 5K. And we get around the approximately same kind of genome recovery from it. With the 58K, the genome recovery is coming around 93%, and with 5K, the genome recovery is coming around the 91%. So it's almost same. So with that, it's provide the uniform genome recovery. And we also perform the rough genetic mapping and the QT analysis using this assay. And these two studies are already published for iron and GCAN-free dormancy utilizing this genotyping assay. We also started the flagship initiative with the ICRISAT ground gene bank, and we have a plan to be sequence genotype around 15,000 available accessions in the available in the gene bank of ICRISAT. So this initiative is ongoing. We already extracted the DNA for that, and most probably it will be completed by 2026. So what is the need of why 2K SNP passive 2.5K, that 5K is available? So problem is with the 5K say that it's based only on the 30 cultivated genotype, mostly the Virginia runner. And the DNA isolation requirement, which hampers the accessibility by our ESA and the WCA breeding programs, because they don't have a facility to be for the extractions and sending the sample to other place. So sometimes it's little edits for that. So this EIB had offered the support for groundnut in 2020, and the reference genome of tetraploid is already available. So with that, we have the sequencing data of around 264 cultivated genotypes. So we selected the gene markers from that, and we the markers which are showing the high PIC, less heterozygosity, and those markers we are just provided for designing of this assay. And the good thing is that this assay provide there is no need of extracting DNA. You have to just punch the sample, send to the DART facility available in Sweden, and you will get your result in next 15 days. And no need of the expertise of bioinformatics. So this assay is available with the 2,500 markers, including the trait-specific markers also. So if you want to be track the favorable alleles, all those things, you will get the things done with that one. There is no need to be send the samples differently to a different place and wait for the long procedures. And this final assay is validated and but not deployed till yet for long runs. But we had some genotypes coming from WCA and the ESA. So we had just make some diversity analysis, just try to make the validation of that one. And we also provide it will provide the boost for the ESA and the WCA breeding program in groundnut. So we have another low density marker genotyping for assays, which are selected from the different studies. And these markers are be used for the routine analysis of hybridity confirmation and the quality check and all those from. So these SNPs are available at the Intertech platform, and these are successfully deployed in Asia, WCA and ESA groundnut breeding program, and also developing a 3 major breeding program in India also with our partners in ICR DGR, US Dharwad, and RAR's Tirupati. So these are the assays available. I just give the overview. What are the genomic resources and technologies? So albino diagnostic marker, which are linked with the gene of interest, and we develop the markers from that. So this is just a overview of that, how we can select the markers, polymorphic markers can select. After that, we develop a genetic map, perform the QTL analysis, perform genome-wide analysis, and the marker which is directly linked to our trait of interest. We select that one and validate that one. So we had selected the trait markers. So these are the traits. These are the traits which are be targeted, and these are the traits which are targeted, and these are the cast passes are available at the Intertech platform. And other new traits we also included. We have the lead in those traits also for the high iron deficiency tolerance, form weight, and we are working on the salinity tolerance and the sugar content also. So this is the marker development and deployment roadmap at ICRISAT. So we are in the scoping of trait and trait discovery. So we already completed the marker validation for these traits, and the results came last week only. So we didn't include that. We just validate the markers, but the deployment is still yet pending for this one. But we have now marker development available for the markers like rust resistance, LLS, high expressive dormancy, and blanchability. And we also have the markers of breeding release varieties also with our NARS partners from ICR DGR with Dr. Vera, and from USSR from Dr. Motagi and Dr. Ramesh Bhatt. So these markers are available at the STPG under the STPG project at Intertech platform. And these are the markers which are used across the globe for the deployment. And this is the overview of that. So my take-home message is that genomic resources are cost-effective, and it's provide a high-throughput genotyping, genetic, and software. So it's not a no more issue with that. So we require now the precise high throughput phenotyping. So it's a still challenge in addressing key climate change threats, and the integration of next technology, next generation will ensure developing the improved crop varieties faster with the more precision, and our international, national and state government, private sector support essential for the research and development programs to improve at national and the international agriculture. So I want to acknowledge our partners from across the globe here. And thank you. Here I want to acknowledge my group. Yeah, thank you.
Thank you, Vinay. So can we open it up for questions? Yeah. Can introduce and then ask.
Hey, I'm Samir. It's really impressive how much of the work you guys have done in terms of genomics and advancement of the technology, and I'm really glad I'm here to see that. So my question is that if we zoom out a little bit and then think about in terms of classic equation of genetic gain, so how much do you think that has been improved with the technology you have used in terms of getting the genetic gain over time?
That's a good question. But right now, because the availability, this is a tetraploid crop, and we have that tetraploid reference genome is available in 2019 only. So we had now in that stage, we had a lead in for the few traits, and we are also validated markers are also available at different platforms, and it's routinely used for different breeding program also. If you are specific for the genetic gain, it depends on how it can be utilized. It is just application. We are just providing a boost to the breeding program. So it can help you out in the things you will get in 5 years. It can help you to just done in 2 years. So it just make your things shorter. So it be a benefit maybe depend on the later to the genetic game. I'm not sure how successful it can deployed in the coming years. So I'm unable to answer genetic gain and this stuff.
I mean, that's enough. Thank you very much for a nice talk.
Yeah, thank you.
Yeah.
Hi, Vinay.
Yes, sir.
Nice talk. My question is regarding the updating of the panels.
Yep.
The 2.5K panel that
Okay,
is with
sir.
the EIB.
Mhm.
So the new traits are emerging up, right? Like seed dormancy, blanching, and all. So how you are planning to integrate those markers into this 2.5K
Sir,
panel?
with that 2.5K, as I mentioned, the problem with the 2.5K, the markers are fixed. We can't include the other recently validator markers in that one. So we have only the markers available only for the LLS, rust, and fresh dormancy, seed weight, and these 6 traits and the QC markers are available. And we have now validated markers for blanchability, high iron and zinc, and all those things. So we have the opportunity to include those markers at Thermo Fisher platform and the markers is with 5K. So we have a plan to include those validated markers at the Thermo Fisher platform because it's not be feasible with the 2.5K. The cost is coming same around that, same for the 5K and the 2.5K. But the thing is that the prerequisite is the DNA. Yeah.
The concern here is the updating of the markers for the traits into the panel.
So that's not feasible with the 2.5K actually, sir.
So we need to go with the low density panels only. But if we want to
It depends upon your requirement actually, sir. What you are actually targeting. We also have a trait specific around 178 trait specific markers available at the Intertech platform. So if you are specific, you made a panel of 10 SNPs, just plan accordingly and just send the samples. Based on that, you just plan your 4 plates and send sample to so it can provide data with that.
Yeah, that's okay, fine.
Yeah.
If it is integrated in 2.5, that will
It's not feasible. Actually, it's a fixed assay.
That's
But it's
fine.
feasible on the 5K assay, sir. So we have a plan to include the recently validated markers on 5K assay in the coming days.
Yeah. Any more questions? If not, so I have two comments. So one is the second one, which Mohan has raised. And Vinay, I think probably the need is to keep on updating this 2.5K assay with the new trait-specific markers. That would be
Sorry, ma'am,
useful
not a
for you.
2.5K, 5K assay, because 2.5K assay is fixed. So whatever the markers recently validated, we have planned to include that one on the 5K, which is available at the Thermo Fisher platform. Yeah.
No problem. In any of the one.
Yeah, yes,
platform, so
ma'am.
that
Yeah,
would
we
be-
can include that one. Yes, yeah.
Thank you so much. And then again, pinning on that important question which Samir has raised and how the genomic tools have contributed to increase the genetic gain. You or from your team member can just think about which of the component of the 4 components of the genetic gain equation can this genomic tools can really contribute.
Yeah, so it can provide help here in the early generation selection. So and find the variability. So with that,
So anyone in the group want to attempt to answer that question? How do we use these markers? You want to? Please go ahead. Can you give the mic there?
For the house, my inputs like I am Dhaneshwar Deshmukh from DCM Serum. So the moment we think of deploying genetic tools into breeding program, there we increase the selection accuracy of the individuals which we are going to have it into population, number one. And its selection intensity is going to be increased because of the accuracy of the markers into breeding program. Rest all parts can be taken care of operational, say, breeding cycle, but these are the 2 most important contributions of the breeding tools. Thank you.
Thank you.
So thank you, Dhaneshwar. And then Dr. Bhatt wants to add something. Yeah. So fine. So thank you. So I think so selection intensity from our work experience as well. So selection intensity is the one. So because genomic tools, particularly with the marker arrested selection, would enable us to select from a large selection
Yeah,
population.
it provides early generation
Thank you
selection
so
also.
much for
Yeah,
that
thank you.
wonderful presentation, Vinay.
Thank you, ma'am. Thank you.
And then this is a good start for this topic on genomics assisted breeding. And he has summarized to us all set of tools which are available for using in the breeding. Thank you so much.
Thank you.
Please give
Thank
him
you
a
for-
big round of applause.
Right. Now I request the second speaker, James Mololo. He's a groundnut breeder stationed at ICRISAT, Malawi. I've been working him for the last 3-4 years. His talk today will be groundnut breeding advancements, efforts towards genomic selection for quicker genetic gains.
Good afternoon, ladies and gentlemen, as well as my English scientists. I stand here today, this afternoon, to share with you some of the big highlights in relation to genomic selection and what we are doing in efforts to ensure that by end of the day, we have an impact to smallholder farmers like the ones shown in that picture. I thank the previous speaker, also kind of tried to read my mind in the first slides about what are the key challenges and how can we harness innovations as well as technologies to be able to move forward and ensure impact is enhanced on the ground by end of the day. For the outline of my presentation, first gives the context and the challenges and the opportunities, long-term technical approaches, then the use of genomic tools as a game changer. Then I'll go quickly to association map and the genomic prediction by modeling, or to highlight what we have done in relation to this. So we all know that agriculture is facing a perfect storm due to emerging challenges, the case of climate change, emerging new diseases, increased food demand, because even come 2063, we'll have millions of people to feed. And at the same time, there is the need for supply of quality food so that we can be able to feed the population. And at the same time, the need to adapt to this dynamic environment whereby we need technology or technological advancements to drive forward new collaborations so that together we can be able to deliver. And of course, smart strategic planning grounded on objective view of the current data state and a data-driven analysis of market opportunities and expert insights into future scenarios would be key if we are going to remain competitive and be able to outcompete these emerging challenges. Of great importance in the region is biotic as well as abiotic stresses. So biotic be very major key. For example, the groundnut rosette disease, the leaf spots, the issue of aflatoxin as well as rust, which is kind of an emerging and rapid expanding disease within Africa. So a long-term approach in relation to address some of the key challenges whereby we need efficient as well as effective and impact-oriented efforts by ensuring that we exploit the existing opportunities. How can we harness innovations and the technological advancements to address issues such as adaptation and resilience to be able to meet the food as well as nutrition security needs? And of course, this would only be driven by strategic partnerships coupled with strengthening our institutions so that together we can be able to use that as a vehicle for delivery. So genomic selection enhancement of breeding efficiency. So when the question was asked about the use of genomic selection in enhancing genetic gain, again, I said, I'll not preempt this, but it is highlighted here. For example, the use of genomic selection would be able, one, to speed our progress in selection. And at the same time, it will help us also in cutting costs, because by end of the day, since we have generated genomic selection models based on phenotypic as well as genotypic data, it means that in the near future, the animals resources that are being used for phenotyping will be reduced. So in my presentation, I say that genomic selection by end of the day is going to enhance my breeding pipeline management in relation to increasing the number of advanced products that are going to get out of the system and then being delivered to the smallholder farmers in terms of improved varieties by end of the day. So to set the scene of my presentation, I just wanted to quickly highlight some of the phenotyping approaches that as some breeding program within ICRISAT we have optimized. For example, the phenotype for the groundnut rosette disease, early leaf spot diseases, as well as rust screening. This means, honestly, these optimized approaches or protocols would ensure that we generate the quality data that by end of the day will help us in coming up with the right molecular markers, the right genomic prediction models that are going to be fit for purpose. And by end of the day, ensure that they reduce our burden of the intensive screening processes on the ground. So thanks to Dr. Ningam, because he was in Malawi and is one of the key persons who enhanced or who supported in development of these initial tools. And by today, we are able to show in the market some of the resistant varieties, for example, to groundnut rosette disease, whereby we have used these protocols and we have delivered that. And then now it's the high time that we harness the advancement in technologies so that we can be able to move with speed. In addition, we have also accomplished studies on vector-host interaction, as well as the development of molecular markers to support GRD work. So this is await validation. Then also Africa core-set genotype and genome-wide association analysis for associated key traits also done. Studies on prediction models, build on studies from earlier efforts still to be done. Then high-index genotyping and the prediction accuracy for different genomic selection models and the cross-validation approaches for key traits have also been done, and more need to be done so that by end of the day, we have refined models, refined tools that will help us move forward. So going deeper or changing the tune a bit to what I'm going to present today in relation to genomic selection, first I'll give highlights on association mapping work, which we did using cross-country data sets, that's from Malawi as well as Uganda. Then assess the performance of genomic prediction models based on phenotypic information. So we did cross-country evaluations of advanced breeding lines, 192, that's in Malawi and Uganda, 2 sites in each country. Of course, you see experimental design of lattice given the large numbers of the lines, then adaptability as well as stability of the different genotypes was also assessed or analyzed. Then for the genotyping work, leaf sampling, DNA extraction, and genotyping were done using standard procedures. So say then don't need to go to details because we all know the standard protocols that are in place and that we have been using as a routine. Then the genotypic data was generated using scheme sequencing that led to the calling of that 9,000 SNPs, from which filtering was done. And by end of the day, we ended up with 25,101 high quality SNPs that were retained and used further for the GWAS analysis. That's the genome-wide association mapping analysis. Second, in relation to the development of the prediction models, phenotypic analysis were performed in 2 stages. One was the calculation of the best linear and biased prediction loops of each genotype at the environment. You see the R statistical programming. Then secondly, the loops obtained in the first stage were used to fit 3 different prediction models. That is M1, M2, and M3. M1 is whereby it only considered the environment plus the genotype, that's the lines. Then the second one incorporated the environment, the lines, as well as the genomic data. Then the third one incorporated environment, line, plus the mark data plus genomic times environment interaction. Then the predictive ability of the 3 models were further evaluated under 4 cross-validation schemes. That's CV01, CV0, CV00, CV1, and the CV2 that stimulates the realistic problems faced by breeders at their different stages of their work. So for the cross-validations, the CV2 predicts lines tested in sparse multi-location trials. So here again, we wanted to access, is it possible to conduct sparse trials that by end of the day will be beneficial to our breeding program? Then the CV2 predicts performance of new developed lines that have been observed in any of the tested environments. But there is available phenotypic information from other lines in this environment. The CV0 predicts test-led lines in an observed environment by leaving one environment out. Then CV00 is similar to CV0, except that it also considers the prediction of novel genotypes that have not yet been tested in any environment. And for the model assessment, a random 5-fold partitioning of the entire population was used, CV1 and the CV2, while for CV0 and the CV00, the correlations between observed and predicted values were computed for the leave one environment out scenario as described by Professor Eta. And all the prediction models were fitted to see the Bayesian general linear regression package that implements a large collection of Bayesian regression models. Then, of course, in the SNP utility involved removing all markers that had more than 20% missed value and a minor allele frequency of below 5%. Then SNPs with a core rate lower than 80% and heterozygosity larger than 95% were also excluded, and the final retained genotypes contain that 8,000 SNPs. So for the first study in relation to evaluation of genotypes, looking at the stability as well as the association mapping, one, we observe significant variation across genotypes for all the yield-related traits within and across locations. Then, of course, the three of the sites, that's Nakambago in Uganda, Chiteze in Malawi, and Serele were clustered as one mega environment with 5 more stable genotypes as indicated. So that means, again, these 3 environments being grouped together, we can be able to narrow down and cut on cost and use, for example, one environment, since it seems to be inclusive, and it can be able to be used to serve as a representative of the 3. Then principal component analysis revealed that markers are retained were of high quality, with the first 2 principal components explaining 61.5% of genetic variation. So again, this means these are high quality markers that can be considered for further use in, for example, MDP, as well as development of QC markers. Then the population structure analysis cluster the genotypes in 3 distinct groups based on market classes. This is basically the Virginias, the Spanish, as well as the Valencia. Then 4 significant association signals for leaves for resistance on chromosome 7 and 18 were detected, as well as 8 and the 4 marked trait association recorded for days to flowerly and yield respectively. And one of the marked trait association for days to flowerly was colocalized within uncharacterized protein, while another one was consistent across the 2 countries. And of course, the identification of this chromosome also concurs to some of the studies that have been done, for example, by Manish, Zhang Eta, and means that, again, we're on track. Then future studies are needed for further refining of these molecular markers. Variance components in relation to the prediction models, the addition of interaction between genomic and environment effects reduce the variability captured by the environment, as well as the residual terms, as shown in this figure, whereby, for example, the red shows environment, genomic is green. Yeah. Then the main effects of environments consistently explained most of the total variance for all traits, as we all expect. And the most complex model, that's model 3, that included the environment, the lines, the mark data, as well as the G/E at the lowest environmental variance, while the M1 at the highest in all the 4 traits. Then model M3 explained the lowest residual variance in all the traits. Again, showing that that more complex model would be more ideal. Then as I go towards YD up, the predictive ability, all the 3 models had higher predictive ability, and a CV2 for all the traits compared to the other schemes. So again, the CV2, where we said we combined the environment, the lines, the marker data, as well as GE. This seems to be the best. Thus, moving forward, it would be the best to use for further development of prediction models or for genomic selection work. Under CV2, the average predictive ability was 0.6 to 0.68 for yield, then 0.26 to 0.004, especially percentage, that means it's very low, 0.5 to 0.59 for seed weight, as well as 0.79 to 0.86 for 100 seed weight. And of course, the CP1, the VA range was 0.2 to 0.51 for yield. For Shelly percent, 0.08 to 0.35, again, seems to be very low. Then 0.1 to 0.47 for seed weight, and the 0.05 to 0.47 for 100 seed weight. Then in CV0, the PA range, again, is as indicated. And of great importance to note is that in the CV00, the PA range was the lowest. That this is not one of the best cross-validation approach to use. In all the four schemes, including market data, improved the predictive ability. So that means when we are developing these prediction models, the market data is key. We are to make some meaningful progress. Then incorporating GBI in M3 model improved the predictive ability in CV2 and the CV1 schemes and reduced the residue and environmental variance. Then the high PA in CV2 implies that space testing could be implemented, while the low PA recorded CV00 shows that it is difficult to predict untested lines in new environment. So as a conclusion, the findings on market trace association pave way for marker-assisted breeding and for genomic selection for earliness and yield. And the markers detected as most informative can be incorporated into mean density panel for potential use in future genomic selection, as well as for the development of QC markers. First testing, of course, we confirm that it is possible to be implemented in our ground breeding program. Then the GS has a significant potential to increase genetic gains and improve environmental variance modeling. So finally, I acknowledge the team we have been working together. So this is a study that involves over 10 member teams from Makerere University, from within ICRISAT, from Nasari in Uganda. Then of course, we can't ignore or overlook the support, the financial support from the International Treaty on Plant Genetic Resources for Food and Agriculture through the Benefit Sharing Funds, the Regional Universities Forum for Capacity Building in Agriculture, as well as the funding or support of the students under the Academic Mobility Scheme, and of course, some support from the Peanut Innovation Lab. Those are my few submissions for today. Thank you, team.
Yes, Charles.
Hi, it's a good talk. I have a question about I saw the slides you're doing the variance component analysis. Most of the for the environmental variance is ranging from the 50% to 80%. So that's a big variation on the environment. What do you think that is feasible for the breeder to use that model to do the genomic selection? My second question is how are you going to reduce that environment variance to improve the accuracy of your model? Thank you.
I think I did not get the first question well, please.
First question is your variance component analysis. Most of the variance environment is ranging from the 50 to 80%. So you had the slides. So I was wondering in that big range for the environmental variance, how are you going to use the in your
huge environmental variance, which you have shown in the slides, right? How does that affect the model accuracy? And what is your strategy to going forward?
Yes.
Okay. Thank you for that question. We all know that, for example, when we are doing modeling, environment is key, and variation is across. Because when you look at the results, you find that the genotypes, the variations within the genotypes is not that much. But environment is wide. And that's why, again, we are bringing in the aspect of the G by E interaction, because we cannot leave that out. And that means that if we are to get authentic, conclusive results, one, we need, again, for me to do further phenotypings, as well as further refining of those molecular markers, again, to make a concrete decision. Because, for example, this was the first study. And we are saying that this forms a basis for us to do more further screening as well, refining of the molecular markers. So in the next 1-2 years, again, we are putting more resources together to ensure that we generate, again, more robust phenotypic data across different environment, adding more locations, more countries, and also bringing into play the wide scope of the molecular markers to be able to see if by end of the day we reduce that gap.
Actually, I have one point when where it will answer some of the questions. It will give some answer to the Charles point, and also it will give some inputs to the our speaker. First, because in any multi-environment testing, environment usually plays a huge role, because as the number of environment increases, the percent contribution by the environment has a greater role, followed by G by E, and then where genotypic contribution by per se, it will be very low as the number of environment increases. So in order to overcome this, we can use the concept, there is a new concept called envirotyping, wherein they integrate the phenotypic data, genotypic data, and also environmental data into the analysis. And then they perform the G by E, they can do the G by E and identify the, they can make a precise analysis for their genotyping. So they can use envirotyping. Yeah. And then my second point is with respect to your M3 model. I think already in 2018 and all, we had a similar set of concept, wherein we used, because earlier stability model, they used, when we had a different location and then years, we used to combinedly study it as an environment. Instead, we proposed a model where we can study the location effect separately, year effect separately, and their freeway interaction separately. So similar such type model we have also proposed, and it's already published. Maybe this may be helpful.
Yeah. No, thank you for that. I think he answers some of a lot of questions. So we'll have a discussion. I think I buy in that idea of the location, the specific location prevailing conditions is very key if we are to get that.
I think I have a similar presentation tomorrow. I'll be making a detail of.
Thank you. Thank you. Yeah.
Thank you, James, particularly highlighting on some of the phenotyping tools. And then thanks, Ajay, for bringing that idea of envirotyping. So we have been always putting this environmental variance as just one component, as something unknown. But I think it's time that we dissect it and see which of that environmental component is contributing to how much variance, and then design the models accordingly. Thank you. Over to you.
Thank you. Moving forward, the next one is a presentation by Dr. Zhongzi Zhao. His topic will be dissecting of phenotypic variability and identification of allied genes loci in cultivated wild Arachis combination will population. It's online.
Thank you. Thank you.
Go ahead, please.
Yeah. Can you hear my voice?
Yeah.
Okay. So good afternoon for everyone. My name is Chuanzhi Zhao from Shandong Academy of Agricultural Science. due to the weather reason, I'm unable to attend the conference. First, I want to express my thanks to Dr. Manish for this invitation and for organizing these amazing conferences, and also providing me the opportunity to present my research. Today, my presentation will focus on dissertation of phenotyping variation and identification of elite genes and losses in cultivated wild Eritrean repopulations. In China, peanut presented one of the most important oil and cash crops. Also, in China, peanut is usually called longevity fruit because of their many healthy elements. A study published in New England Journal of Medicine reported that eating peanut daily can reduce the death from heart disease by 29%. And even eating peanuts just twice a week can reduce the risk by 24%. So please remember to eat a peanut for everyone and everyone behind our friend. However, in China, peanut production are facing many stresses, which are seriously threats to peanut production, especially for late leaf spot disease. When it occurs, the yield is generally reduced by 20%, and in certain cases, the reduce can exceeding 50%. So breeding resistant peanut varieties is the most important way to control this disease. As we know, that cultivated peanut is tetraploid. It derived from the hybridization between Deonosis and Apenensis, and the genetic background of peanut is very narrow. And the most cultivated species are sensitive to iOS. For example, in China, about 70% of the peanut cultivators come from two elite germplasm, 石头旗 and 浮花生. And we know that there are more than 70 wild species of the peanut, and some of these wild species are with high resistance to the LAS and many other diseases. And this slide shows the resistance of several wild species to the main diseases of peanut. And we can find that the most of the wild peanuts are with strong resistance to the main disease of peanut, and some of them have high resistance and even immunity to LLS. These wild species are valuable resources for identifying the resistant genes and improving the resistance of the cultivated peanut. However, cultivated peanut are tetraploid, while most of these wild species are diploid. So the for these differences, eliminated the utilization of these wild species. So to using the resistant genes and many other traits of the wild species, we obtained an artificial tetraploid of the wild peanut, IPDUR, with the help of the Dr. Solaya and Dr. David from UGA. And using several SSR markers, we confirmed that the specific bonds, and this picture shows that some specific bonds could be from the Deonosis and Apenensis could be detected in the new germplasm IPDUR. Meanwhile, the genomic size is also the sum of the duonesis and the epinesis, and similar with the tetraploid genomic size. So we confirm that this is the real artificial tetraploid material. Then using this artificial tetraploid IPDOR as the parent to cross with the cultivated peanut T-front, we constructed TI population. As we know, T-front is the first cultivated peanut varieties for the whole genome resequencing research. And yesterday, Dr. Bao Zhuguo provide a report and tell us why they choose the T-front as the peanut variety for whole genome resequencing. And we found that the TI population showed high, huge variations in several traits, including the resistance to the LLS, to the ELS, and the seed size, pod size, seed number per pod, pod number per plant, lateral branch angle, and the number of branches. For example, some real lines can produce more than 200 pods per plant. And in this slide show 2 pictures from 2017 and 2018. We can know that some lines have a huge biomass, and the lateral branch is more than 1 meter. And the disease resistance of the parent and the real lines of the TI population were detected, and the IPDOR showed significantly high resistance to LAS than the female parent T-front in for environmental test, and the LS score displayed a normal distribution, suggesting that the resistance were controlled by QTLs. Then we selected 200 lines of the TI population for whole genome resequencing. In totally, we obtained about 2.77 TB clean data. And using the polymorphic SNPs, high density beam map was constructed, which covering about 2,050 city Morgan, and our average interval between two markers was about 0.43 city Morgan. In totally, 28 QTLs associated with the LLS resistance were identified. Among them, the major QTL LLS 14.1 could be detected in all the 4 environmentals. And this QTL could be detected stably in 4 environmentals. In addition, the other QTL LLS 3 and LLS 14 were also detected in at least 3 environmentals. Bioinformatic analysis showed that to the major QTL LLS 14.1 and 14.2 were mapped on 2.58 city Morgan region or LG 14. Interestingly, we found that almost all these SNPs in this interval originated from the IPDOR. This result indicated that the existing large no exchange segments in this interval, and the resistance genes of this region come from the wild speech IPDOR. And interestingly, within this genomic region of this QTL, a total of 28 disease-resistant proteins were identified, and most of them are TIR, NBS, LR genes. Among them, 6 R genes showed a significantly sequence difference between the Tiflander and the wild species. We predicted that the 6 genes might be the candidate genes controlling the resistance of the IPDOR. Also, haplotype analysis showed that 2B markers C14M65 and C14M86 were closely related with the resistance in these 200 lines, suggesting that the 2 markers were useful in microassistant breeding for improving the resistance of the peanut. Previously study identified several QTLs from chromosome 3 and chromosome 13. In our study, we confirmed that these QTLs interesting this the resistance of the previously identified QTLs come from the chromosome 4, chromosome 3 and chromosome 13 might come from 2 germplasm GP NCWS 16 and GPB D4. Interestingly, these 2 materials also come from the introgrowth of the wild species. And the detailed result of this work have been published in TEG, which can be downloaded. In addition, using these populations, we identified many QTLs for other traits. For example, in totally 12 QTLs for early leaf spot resistance were identified. We also identified a major QTL for the pod side from the chromosome 5 and predict 2 candidate genes. And in totally, we identified 10 QTLs for the branch length. And we found that the branch length of the female parents of the Tiflander in different environmentals range from about 45 centimeters, while that of the male parent IPDOR is about the branch length is more than 100 centimeters. So there is a significant differences between the 2 parents. It was observed that the lateral branch length in the real lines showed a huge variation. In 3 environmentals, the minimal value of the lateral branch length is about only 18 centimeters, and the max value is about 220 meters. And also the branch length showed a normal distribution. And we we identified 6 genes for the lateral branch length. And for the main stem height, a significant difference was also observed between the 2 parents. And for these traits, we identified 3 QTLs associated with the main stem height, and with the latest one accounting for 9.5% of the phenotyping variation. The number of branches is one of the most important traits in peanut, which is associated with the biomass and yield. We observed that some lines pose only 2 branches, and for example, for this line, and some lines have more than 20 branches. Such a huge phenotyping variation enable us to identify 9 QTLs related with the number of branches. And finally, I'd like to thank to share our recent progress in the genome assembly of the wide space lattice globata. Globata is recognized as one of the most resistant wild peanut varieties. And we have completed the paper and we will submit it in the future. And last, thanks to Dr. Xingjun Wang, Dr. Jiao Wenpan, and Dr. Han Xia from Shandong Academy of Cultural Science. And also I want to thank to Dr. Manish or EcoSet, thank to Dr. David Saraya from the UGA and Dr. Liang Qunhe from Guangxi Academy of Cultural Science, and for their help for creating the new materials and the phenotyping identification. That's all. Thank you. Any question?
Thank you. Thank you, Zhao. Any questions for Zhao? Charles, go ahead.
Hi, Dr. It's a good presentation. I see the slides on the QTL identification for the main stem high. I realized the LOD is 2.3, and then you claim those 3 QTL. My question is, what are your justification to claim those 3 QTL since the LOD is a little low? Only like a 2.12, the highest only 2.6. So I was wonder how you can claim that QTL. Thank you.
Thank you. Thank you. Thank you for Professor Chen. It's a good question. Yeah, and we identify the QTLs according the cutoff of the PVE is PVE more than 5. Yeah. For the QTLs of the MHS, the LAD is very low. Yes. Thank you. Thank you.
Thank you, Zo, for that nice presentation. Extensive phenotyping of 400 plus RILs. Can you just share what are the experimental designs you have used for assessing them?
Sorry, I think I didn't catch you. Which phenotype?
Yeah, for phenotyping of your 400 recombinant inbred line populations, we have done it in the field. So which experimental design have you used?
I'm sorry.
So no problem. Thank you for that.
Yeah. Okay. Thank you.
But that's just for the advantage of the group. Just want to mention that in the previous presentation, when James has presented, when we had a population of 200 plus, he has used alpha lattice design, which is good. Because as one of the editors of the tag, I really struggle with several papers coming with 400 plus populations and randomized complete block design. Thank you.
Thank you.
Thank you, Zhao. Appreciate it.
Thank you. All right.
All right, moving forward. Our next presenter is Dr. Kenta Hirosawa. His talk will be pyramiding of flavor quality, disease resistance, and high oleic content through genome-wide SNP genotyping in peanut.
Okay, so thank you for your kind introduction, and I'd like to thank for the organizers to provide this opportunity for our activities. So my presentation is a kind of a trial to pyramid multiple traits in a new cultivar. So my name is Kent Shirasawa from Kazusa DNA Research Institute, Japan, and this project is collaborating work with Chiba Prefecture Agriculture Forest Research Center. So Chiba is a name of the prefecture of Japan, and our institute is also located in Chiba. So first of all, I'd like to introduce our institute for you. So actually, our institute is focusing on plant genome sequencing. So since 2000, with Arabidopsis, we released many plant genome data, mainly crop species. One of them is peanut. So peanut is actually not so important crop in Japan. So as you can see here, production amount of peanuts in Japan is not so big. Japan is ranked in 59th place, like here. However, peanut is a very important crop for Chiba. Actually, Chiba is a very famous prefecture for peanuts in Japan, and our prefecture produce more than 80% of production of total amount from Japan. So that's why our institute and the Chiba breeding station are collaborating to develop a new cultivar. So this is the most famous and the most popular peanuts cultivar in Japan, namely Chiba Handachi. So this is a very old cultivar bred in 1952. However, at this moment, this is the highest quality cultivar. So that's why the price is also the highest. So to overcome this situation, the Chiba breeding station try to develop a new cultivars. So this picture indicates a new cultivar released within these 5 or 10 years. So the left side, you can see a new cultivar, namely Q-nut. So this cultivar has a high sugar content in the seed, and this name Q-nut is a name after a kind of next generation peanut, because next letter of P is Q. So the breeding station named this cultivar as Q-nut. The right side panel indicates another cultivar, Chiba P121. This is a high oleic acid cultivar, and this cultivar are developed by collaboration work, the breeding station and our institute, based on the genomics based breeding technologies. So in this under this station, we try to develop new cultivars. So this is our next goal to develop a new cultivar by pyramiding 3 phenotypes. So 3 phenotypes includes flavor quality, high oleic acid, and hot root resistance. So flavor quality should be came from Chiba Handachi, and the high oleic come from Chiba P121. And the disease resistance, we will use Thorson this is a US cultivar and known as a resistant to pod rot disease. So to establish the pyramiding line, we crossed Chiba P121 with Chiba Handachi, and the resultant F1 are crossed with Torson. So now we are obtained F1 populations, including 130 individuals. So based on this population, we have to establish new cultivars. So in this breeding program, we will use DNA marker. However, only a high oleic acid can be selected by DNA marker technologies. However, flavor quality and disease resistance, we have no information on the DNA markers. So we need to develop DNA markers for these two traits. So this is a strategy how we can generate, we can develop DNA marker for the two traits. So the first step is identification of genome-wide SNPs. The second is genotyping of the selected SNPs from the SNP discovery. And the final step is the linkage analysis to identify, to find a genetic loci to control the two traits. So the first result, we employed DD-RAD-seq technologies because this technology is very efficient to develop DNA marker over the genome. And this protocol is already established in our institute. And we applied this technology for many types of crops. However, in case of peanuts, this technology doesn't work because as you can see here, number of SNPs obtained from DD-RAD-seq is very limited, like 10 or less than 10 SNPs were available for every chromosome. So to overcome this situation, we switched the technology to whole genome shotgun sequencing. So based on the result of the WGS, we could obtain 20,000 SNPs every chromosome. So based on this information, we moved forward for genotyping analysis. Actually, we developed a protocol for target amplicon sequencing. That means we can select SNPs from the genome, and we design a PCR primer to amplify the SNP loci. And the amplicon should be sequenced with NGS. However, at the moment in 2016, in this protocol, this is a kind of handmade. So that means we designed a primer one by one by manual. So that's why the number of SNPs are very limited, like less than 300. So we now just change the technology to employ a Twist Bioscience kit. This kit is very useful or very helpful to handle 96 samples at once. So the DNA extracted from 96 samples were fragmented by enzymatic method, and the fragmented DNA are cooled by coating. So we generate we generated a ultra high throughput library from this 96 samples. So after that, we could enrich, we can capture the SNP DNA fragment, including SNP loci. So using the probe, capture probes, we enriched the DNA fragment by hybridization technology to establish the enriched libraries. So these libraries are sequenced on a short read sequencer, namely MGI DNAB6, and we obtained 90 gigabase data from the 96 samples. So this is a result of the genotyping analysis. We designed more than 6,000 SNPs over the genome, and the result from the sequence data, we could genotype around 6,000 SNPs. That means most of all designed SNPs were successfully genotyped. So the cost is a little bit high. That was $30 for samples, but this is a first trial, so the situation will be improved next time. Anyway, we could discover the genome-wide SNPs, and we could genotype the genome-wide SNP loci across the population. So the final step is a linkage analysis. So to perform linkage analysis, we need phenotype data. Unfortunately, phenotyping is still ongoing, so I'm sorry I can't present the result of phenotyping. Anyway, I'd like to conclude my presentation. So genome-wide SNP genotyping is available with whole genome sequencing and target amplicon sequencing technologies. So once phenotyping is completed, linkage analysis will be done to identify genetic loci for the trait or to develop DNA markers. So using these technologies, we believe we could breed high quality cultivar in near future. So I'd like to thank all of the lab members to support this project. Thank you for your attention.
I have one question. What's the duration of the peanuts in your place? How long maturity?
Actually, Chiba Handachi is a kind of late maturity, so we need to develop early maturity cultivars. So that is another big target for our breeding program.
How many days it takes? 140?
More than.
More than that?
Yeah.
Any other questions? Audience? Yeah, Charles.
Thank you. It's an interesting talk. I was wonder, I interested to know how you define the quality tree. From your presentation, you showing like sugar content is one of the three. Besides the sugar content, anything else? I'm interested to know that. Thank you.
Yes. So quality of flavor or quality of oil, we will use GCMS or HPLC to measure the content of the flavor substrate and oil. So we already get the candidate substrate for the flavors, and so we can present next meeting here.
Any other questions from the audience? Thank you.
Okay, thank you.
All right. Our next presenter is Dr. Ramesh Bhatt from US Dharwad. His topic for this afternoon is peanut improvement through genomic assisted breeding for enhanced foliar disease resistance.
Good afternoon. So I'll be talking on peanut improvement through GAB, especially for foliar disease resistance. And for the last couple of days, we have been discussing about marker-assisted breeding or even conventional breeding, those things. And there have been two areas, probably, where GAB has been used extensively and the applications are made and the products are also ready. One is the oleic acid content and second is foliar disease resistance. So in my talk, I'll be talking about the application of GAB, genomic assisted breeding, for improving foliar disease resistance. And I'll be highlighting how and why the applications of GAB have been made for improving foliar disease resistance. At least in Karnataka, where I have been working, lately spot and rust are very important, and they can cause up to 70% yield loss. And the yield loss also varies among the genotypes, starting from, taken in this graph, you can see maybe 17% in GL24 to about 50% loss in SP206 or DH8. And a successful story has been the breeding of GPBD4, which is also known as Vikas. And the hybridization of GPBD4, the first cross was made in 1991 by crossing KRG1 into ICGV 86855. Of course, we have heard so many times, it has got a blood of Arachis cardinacea. And to be fortunate, I made this cross in 1991 as a part of my MSc study. And later in 2002, after so many selections and artificial selections, natural selections, those things, this variety was identified, it was released and registered at NBPGR, and a lot of seed productions was made at our university. And this is early maturing, high pod growth rate, high partitioning coefficient, and shelling outer, and has got desirable pod and kernel features, high oil content, about 50%, then high oleic acid also, a ratio of 1.6, and significantly superior quality of peanut cake due to high protein content and high quality of farm, which is very important as fodder. And if you look at the yield loss of GPBD4 over other varieties, you can see a significant reduction in the yield loss, which is about 12%, as compared to the TME2 or GL24, which is up to 40%. And last year, we made the scientific and economic impact of GPBD4. This is very, very important for the technologies, whatever we develop. So we've made both the scientific as well as economic impact. Scientifically, probably all of you know, most of the genomic studies, transcriptomics, proteomics, metabolomics, or even basic studies where one wanted to study the disease resistance, especially for foliar disease, this has been used as a positive check. And therefore, it has been referenced in over 500 publications worldwide. Apart from that, it also served as one of the parents of 5 released varieties from our own university, that is G252, DH245, GPBD5, DH232, and DBG4, which I'm going to talk now using molecular breeding. And apart from that, it has been serving as one of the parents of several mapping populations for various traits. I've listed some of the traits here. And coming to the impact of economics, which is very important, over the last, say, 16 to 18 years, it has contributed so much economically to the farmers. For this assessment, in fact, I made use this paper, which was published long back, where GPBD4 and TME2 were evaluated every year for 5 years, 2005 to 2009, and each year, 12 farmers grow, or they have grown these two varieties over 4.8 hectares, and totally about 60 farmers over 5 years, where TME2 yield was 21.43 on an average, and GPPT4 was 25.94 with an increase of 21%. And considering this figure as the yardstick, then because our university produced a lot of breeder seeds, certified seeds, foundation seeds like that, and we have the account, how much seed is sold, and we have the receipt book. And from that receipt book, I acquired the data, and I worked out the approximate area covered by GPPT4 and the approximate price. We had the database during those years, and over TME2, which is used as a check, I calculated the exact benefit accrued by GPPT4. And of course, it's a conservative calculation because, you know, it is a self-pollinated crop, and the farmer to farmer spread is not accounted. It is only based on how much certified seed is sold from the university. You may be interested to know the benefit caused by this one is 17.6 billion Indian rupees over almost 16 years. And this development of GPPT4 not only contributed for the scientific as well as economic impact, but also it helped in a lot of genomics assisted breeding. So development of GPPT4 enabled the genomics assisted breeding, especially in terms of developing mapping populations, which I'll be covering now, and development of the marker systems, because in groundnut, good marker systems are really required, or at that point of it was required, and for the mapping work. And these were the different mapping populations where GPPT4 was used as one of the parents, and extensively used was TAG24 into GPPT4 and TG26 into GPPT4, which have been extensively used for mapping lately spot and rust resistance. I'm talking of 2009, when the collaboration took place between Rajiv Varshney and my own mentor, Dr. M.U.C. Gowda. And the marker systems at that point of time available was RFLP, RAPD, SSRs, those things. But the problem was the lack of polymorphism. You take any marker system, the maximum polymorphism was only 4%. So we wanted a marker system which can give higher polymorphic rate. So in that regard, with the help of Dr. Kenta from Japan, we developed a new marker system for groundnut for the first time. It was called as AHT, and we developed about 3,000 markers using whole genome resequencing of 33 genotypes. And also lately, we developed about 43 lakh SNPs and identified some of the polymorphic markers. So these are some of the publications, if you can read, about the development of the AHT markers, and they have been validated. And you can see this is a distribution of those markers across the chromosomes. Of course, there is a lot of variation between the A genome and B genome, and also between the chromosomes. And out of 3,000, about 82.2% were validated when we performed the PCR in the lab. And you can see the polymorphic rate here. On an average across the various mapping populations, the average was almost 20%, almost 5% of, or fivefold more than the normal RFLP or SSR polymorphic rate. And this is a distribution of those CHT markers. And also we developed a lot of SNP markers with the help of KDR, collaboration between KDR and US Dharwad, and where we used the whole genome resequencing data of 179 accessions available at that point of time, and studied the SNPs over 43 lakh in number across the chromosomes. And this was the SNP density over all the 20 chromosomes. And this is, you also could identify some of the rare SNPs. And of course, transversions and the impact of these SNPs were worked out for each and every SNPs. And polymorphic information content, about 46 lakh or 46,000 SNPs had the highest polymorphic information content, indicating that they are very useful for genomic assisted breeding, whether it's mapping or genomic selection, whatever. Okay, so using these markers, because we had these HT markers, SSRs, all those things, then we started the mapping work. And we went for linkage mapping, association mapping, as well as marker breeding later. And this is how we started the mapping. Initially in 2010, you can see only 2 markers were added in that region where we identified the major QTL. And later in 2016, a lot of markers in the same region were accumulated. And it is a kind of refining. So therefore, in fact, this region actually contributed a QTL which had a PVE of 71 to 82%. So it's almost like an oligogene. And also, the work by Bertielli, the paper which came in 2016, in fact, when they analyzed their own sequence in that region, in the QTL, whatever we told, in that region, they found one R gene also. So that gave actually a lot of confidence in our own work. And later, with the help of KDRI, we further refined that region and using a lot of WGRS and DDRS sequencing, using different genotypes, varying for the phenotype, disease resistance and susceptibility. And then we could refine that interval starting from 8.8 MB on A02 chromosome to 2.7 MB. So identifying or refining the QTL region, reducing that area and identifying the genes, all those things were made. And similarly, we also did for another genomic region on A02 as well as A03 chromosome. And finally, we identified about 10 candidate genes on each QTL regions. And these were the markers which were extensively validated before we employed for marker selection using various approaches. And also we studied the DNA methylation, especially in those 10 genes. And we found that those 10 genes were not susceptible for an epigenetic change. However, there were other 2 genes which were showing epialleles between the resistant and susceptible genotypes. And then using all these resources, we actually went for molecular breeding, where we selected DL24 and TME2. I have shown their susceptibility reactions. And we used DPBD4 as a resistant source and ICGV A6699 as another resistant source. And we went for 3 rounds of backcross breeding using marker breeding, and the markers flanking those QTLs on A02 and A03 chromosomes were used. And this is how we went for the background selection and foreground selection in advanced material like that. And we used a lot of RAD sequencing for background selection, and we found almost 99% recovery of the background genome. And they were almost positive with the foreground selection. And also, with the help of ICRISAT, we used those SNP markers, which were found to be linked for disease resistance. And this is how we used a lot of field work at Harvard. You can see a lot of variation for disease resistance. And then this is GPBD4 as well as DBG3. We call it the background, the backcross line of DL24 was called as DBG3, or it was also called as improved GL24, and DBG4 was named as Super TME2. And later we, because in India at least, we have a set of evaluation for the release. So we went ahead with those evaluations, and over almost 8 years, you can see the disease reaction, always DBG3, the maximum reaction was 4 as against the 9 of GL24. Similarly, for DBG4, the reaction was almost 4, the maximum was 4, whereas TME2 always showed 8 or 9 disease reaction as against the control. And also we performed the station trial for about 6 or 7 years, starting from 2014 to 2021. And in all these years, the yield advantage, apart from the disease resistance, was 31% in case of DBG3 and 47% over TME2 in case of DBG4. And also this is the yield advantage across the trials, like station trial, multi-location trial, farm trial, large-scale demonstrations like that. Because these trials are very compulsory before you are releasing the variety. Many of the breeders know it. So across the trials, the yield advantage was 28% for DBG3 and 23% for the DBG4. In fact, my friend asked genomic selection or the genetic gain. And the release of MBBC lines with foliar disease resistance for Zone 8. Zone 8 is a part of Karnataka, and we have released those 2 backcross lines, and they are called as DBG3 and DBG4. And this is how the history of the development and release has taken place. And this is the accession number where we have submitted the seeds to NBPGR, and also the release has been notified by the central government of India. And to conclude the genetic mapping of resistance, which we started in 2009 with a lot of collaborations, and they have been successful, and we could map the QTLs with as high as 72% PVE, and marker development and applications, because markers were very few, so it was necessary to develop the markers. And then we went for breeding through marker breeding, and also it is commercialized. Now maintenance breeding and seed production is being taken up at the university, where our objective is to produce quintals and quintals of seeds so that the farmers can be served with the seeds and get benefited. So maybe Dr. Nigam sir can add to this one. The conventional breeding starts with identifying a problem and ending with solving the problem. And molecular breeding, identifying and understanding a problem, because we spend a lot of time not only in identifying the problem, but also in understanding the problem using various omics strategies, whether it is genomics, transcriptomics, metabolomics, proteomics, all those things. So what is next is very important. And in any case, we cannot replace the breeding work. So therefore, breeding is essential. And these omic studies or omic data will definitely help in realizing the product maybe more precisely and more efficiently. Thank you.
Thank you. Any questions from the audience? Yes, Charles.
Oh, congratulations for your new line going to release. I just interested when you're doing the breeding market selection. What's the base size for that?
Yeah. Population size, we always maintained, say, for example, we combined both breeding and marker selection. It is not, I have to be very frank, it is not exactly the marker breeding. So we went for conventional breeding only, and selection, when we made the selection, then we went for marker selection, foreground selection. So always, in most of these generations, we always had about 500 plants. So there were expertise available at our university. So therefore, we concentrated only on few crosses, and we developed large segregating population, even up to 500 in many cases.
Right.
Sir, what was the actually pod shape, size, and morphology?
Yeah, very good
Is
question.
it because you named the super TFV2 and improved AL24?
The whole objective, of course, you know why GL24 and TME2 are still popular even after 40 years of release. It's just because of their pod and kernel size. So therefore, there is no way that we can deviate from the pod and kernel features of GL24 and TME2. So DBG3 was almost similar to GL24, whereas DBG4 was almost similar to TME2, except for disease risk.
That's good. It's in there. Thank you.
I'm to the question you have asked. I'm hoping Dr. Nigam will share his thoughts, but I have a question. So when we used to select this QTL in the breeding populations, so although the QTL was there, that didn't guarantee us disease resistance of even 6 or so. So we had to always adopt an,
Sorry, I did not.
even if the QTL region is there, it didn't give us to the level of disease scale on a disease scale up to 6. So we have to always adopt a 2-step process.
Yeah.
In the second step, we had to do a phenotyping that would enable us to select the background genome sequences.
Yeah.
So again, going to that polygenic inheritance of this particular trait, how do you see going forward the use of genomic selection for this trait?
Probably, you know, of course, you were also part of the entire study. Unlike other traits, maybe drought or other traits, here, fortunately, the QTLs explained up to 70 or 80% of PV. That was the major advantage. So it is almost, though it is not, we call it as quantitative trait or QTL, but it was behaving like oligo trait. So therefore, the contribution was almost 70 to 80%. The minimum what we noticed was say 60%, not less than that. You can go through our literature. So therefore, it was behaving like a oligo trait. And we concentrated on 2 regions on A02 and A03 using the flanking markers, 4 markers, 2 regions, and transferred simultaneously. So therefore, that assured the transfer of the marker as well as the phenotype.
Right, Dr. Nigam.
get any one time?
Time frame, no, sir. In fact, it took more than because it was the first time using markers breeding. I had some tough time. So therefore, even station trial, if you look at, I conducted station trial for 8 years, almost 6 years. And at the later stage, I was asked to repeat for 1 more year just to gain the confidence. So in terms of time management or saving the time, I did not have any advantage. But now, given a susceptible variety, definitely I can save time because I have learned a lesson.
All right. Thank you. We'll move on to the last one.
Thank you.
Dr. Ashutosh Purohit from ICRISAT. His talk will be Rapid Generation Advancement, RGA in Groundnut, Challenges and Opportunities.
Hello, good afternoon, everyone. As you all look sleepy a bit, so I will also give a rapid presentation on rapid generation and advancement. So my talk is on rapid generation and advancement in groundnut. What are the challenges and what are the opportunities? So as yesterday, Dr. Bauzu, he was mentioning that people who have food in their plate, they have many problems. And people who don't have food, they have only one problem, that is food itself. So we are all here together to fight for a same cause, that is to fight to feed the world. So in a recent study by UN, in the year 2025, more than 300 million people are suffering from acute hunger, and more than 2 million people, especially in a country like African countries, they are at the verge of famine. And more than 7 million people, while we are talking here, they are going to sleep today without food. So who has the answer for this? As a plant scientist, we are all here together. So we all believe that crop improvement holds the key to feed the world. But with the pace that we are progressing today, can we achieve it? That's the question. So we must have to double the pace of genetic gain to unlock the full potential of crop improvement so that we can achieve the target that we have set for ourselves. So we all are very acquainted with the breeder equation that one of my friend also asked a very good question. So to this breeder equation suggests like we have 3 different components in the numerator, that is selection intensity, accuracy, and genetic variance. And in the denominator, we have the generation interval. So what has happened in the past, as one of my colleagues, Samir, was asking, what is the genetic gain in case of groundnut? So in our recent study that we have found that for different traits like yield and kernel weight and 100 seed weight, we have substantial improvement. In our recent study, we have conducted for last 4 decades robust data that we have. I can assure you we have a substantial genetic gain for this trait. But there are many traits for which we still are struggling to have a good genetic gain, especially traits like selling percentage. So, and in many crops like wheat, there is maybe some other crop, there is a yield plateau. So how much is the selection intensity and whatever we are practicing that can help us in achieving this genetic gain is a question. One of my friend James, he gave a very extensive talk on genomic selection. So how this 3 numerator can be benefited with the help of genomic selection so that it can contribute to the genetic gain for different traits. So with all this background, the denominator component, the generation and interval that holds a high potential to give us and helping us in doubling the pace of genetic gain, because if you are able to reduce the generation time for a particular crop, that directly improves the genetic gain for a particular trait. So we believe that breeder has the potential to play a crucial role, to play the center stage in doubling the genetic gain per in groundnut especially. So in the effort to reduce the breeding cycle duration, people like the famous Normal Borlaug, he started the work in maize, and he started the concept of this subtle breeding. So we can take the advantage of growing the crops in off-season nurseries, where we can have the advantage of taking up 2 crops or maybe 3 crops in 1 year instead of having a single crop, so that can directly give us access to advance the generation in a short period of time. So following the building upon the concept that has been built by Borlaug, at Equisat, the all mandate crops, especially like chickpea, pearl millet, sorghum, we are advancing our crop at a double speed, utilizing the concept of subtle breeding for various speed. Here I have given an example of pearl millet subtle breeding. So we are moving our material from Jodhpur to Equisat so that we are saving the time, and we are having an accelerated breeding program. So the next concept that, in the case of subtle breeding, there are some limitations. Like in case of groundnut, we are able to harvest 3 crops per year. So that may not be so much beneficial for a crop like groundnut, and also it involves logistic issues. So there are certain other approaches like double haploid, as Dr. Mera was mentioning the challenges with double haploids, especially in groundnut. There are we have a success story in maize, because in maize we have a lot of inducer stocks that are readily available. But in crop like groundnut, which is a yellow segmental yellow tetraploid crop, is still a challenge to have a good result out of this double haploid. So what the next approach is rapid generation advancement. So coming to the differences that we have in conventional breeding approach, RGA. So when you look into conventional breeding practices, especially in crop like self-pollinated crop like groundnut, when you made your crosses and you move forward from F2 till F6 by following a single cell descent approach, the seed to seed duration from F2 to F6 it takes a huge amount of time, like maybe 5 to 6 years, depending upon the crop and depending upon how many seasons you are able to take per crop. So this is the duration from moving from F2 to F6 to achieve that desired homozygosity quite long. And not just after that, the previous speaker, Dr. Ramesh Bhatt was mentioning how he is struggling with this field testing and variety release, the duration for these two as well. So this is a fixed one, but we have what we have in our hand to reduce this time period. So RGA provides us the option to accelerate the plant growth and development by optimizing and tweaking and turning photoperiod light intensity and temperature, and thereby people already are successful in achieving 4 to 6 generations per year, so that the speed of our breeding approach can be doubled. So the seed to seed duration, while advancing from F2 to F6, it takes only 2 to 3 years or less through a single seed descent approach when we follow the RGA. So this is the time duration we can play around by optimizing a protocol for rapid generation advancement. At Equisat, the breeding team at Equisat, we have standardized a protocol for rapid generation advancement in case of groundnut. So building on the concept of the Leahy group, in which when they harvested the immature spike and put into germination test. So in case of groundnut, we have conducted a pilot experiment and to see and determine which is the earliest stage at which the seed can be harvested, produce a viable seedling. So we have used early and mid and medium maturity groundnut varieties, and we have tested it. We have harvested the early maturing varieties at 60 days, and the late one at 70 days. So you can see, and then we put it for germination test. So we have divided into 2 sets. One, we have tested for germination the freshly harvested kernels, and another set we have dried it for 5 days and tested for germination. But what we have observed, the freshly harvested seeds are not giving a good result, as you can see here. But the dried one, here you can see the germination was quite great. So if you see the duration starting from sowing, the flowering till flowering, 50% flowering, the duration is not much, it's almost similar. like it takes 30 days. But we are for the late maturity one at 70 days. And total time period from seed to seed was like 65 days for short duration and 75 days for the medium or late duration, you can say. So then the protocol that we have standardized, we are utilizing it to accelerate the breeding cycle, and we are gaining 20 to 35 days using early viable seeds. And we are using a semi-controlled polyhouse at Ikrishet, and we are now testing our material as well. So for that, the most important thing is the growing medium and the the platform that we are using. So that we can harvest the a quantity of seed, so that we can advance it for the next generation. So this is a dimension of the trace that we are using over here. And for the growing media, it's normal like alluvial soil, sand, coco peat, and vermicompost. In a ratio of 3:3:1. And after autoclaving that, and as we need to ensure the nutrition for that, so we are basal dose of nutrition we are applying, so that plant can have its optimal growth. So after we do the nipping. It's a practice that we are following, because the source to sink transformation in case of groundnut, if you see, so that has to be taken care of. So in order to reduce the excess vegetative growth, we are nipping the vegetative part at 30 to 35 days after sowing, so that it enhances the pod filling. So that this is a normal temperature that we are following in the this semi-controlled condition, and this is the photo period and the dark period. In this semi-controlled condition, we have fitted, you can see here, the tungsten bulbs and the fan and pads as well. So in the month of December and January, the temperature goes down. We use these bulbs, tungsten bulb, so as to maintain the light intensity and temperature one. And during the summer period, in the month of April and May, we use the water cooler so as to keep the temperature around 35 degrees. Yeah, sorry for that. Thanks. So we are now deploying the this protocol that we have developed in our groundnut breeding program. So while advancing from F2 to F5, so we are advancing to the single seed descent method, and we are utilizing this protocol. And around 8 to 10% of the genotypes that we are advancing from our entire breeding material from F2 to F3 and F3 to F4 annually. So around 10% of our breeding material, we are now we are following this same protocol to advance the generation. So coming to the challenges that we have the rapid generation advancement in groundnut. So groundnut is a day neutral crop. Means the response that we got from crops like wheat and other barley, maybe other crops, but that response of due to the being the day neutral nature of the crop, so meaning is the flowering and the fruiting are not significantly affected by the length of the daylight. That's the big challenge that we have now. The second problem with the protocol that we have worked on is the excision of the immature kernels. It requires, it's a very delicate task. It requires trained human resources, and the ovule, the protection of the ovule is very important, so that it doesn't hamper the further germination. The third important challenge that we are having is the scaling up. Scaling up is a big problem. Like we are now up to only 8 to 10% of our material we are able to advance the generation. But we need to have a better protocol maybe to scale up in a larger scale. So with challenges comes the opportunity that we have in our hand. So because in groundnut till flowering there is not much changes, like it takes around 25 to 30 days. But we are now targeting to reduce the flowering to pod filling duration. That meaning 30 to 60 days period. We are now targeting to reduce that time frame. And then we are also having a clue, thanks to one of our collaborator, that in one of the region in Tamil Nadu, natural condition, the crop is we are being harvested in 75 to 80 days. So we are trying to mimic that weather parameter in our controlled condition, so as we can have a better standardization maybe. Then there are a lot of in the opportunities section. If you see in our breeding pipeline, already we are following this in a single cell descent method. So this RGA protocol can be very handy in developing mapping population like RIL, RIL, MAGIC, or NAM for various mapping studies that we are talking about today. This can also be combined with the genome editing experiments, and also is very helpful in the recycling of the parents to increase the genetic gain. So having all the lessons that we have learned since 2018-19, we have started working on this RGA, and we have our lessons learned from this. So building on all the lessons that we have, again, we are conducting some new ongoing experiments under a controlled environment chamber, where the temperature are being like around 25 to 30 days, humidity is 60 to 70%. And now we are targeting mainly on this photo period, two different sets of photo period with different sets of light intensity we are using. And also we are using the different phenophases of the groundnut, like different stages of groundnut. We are taking that into also account, and we are again trying to make it a better one. So what's the way forward? So yesterday I was talking with Dr. Kuldeep, and he mentioned that the genomic selection has to be integrated with RGA if at all it has to be a success story. So this is a concept that comes also the CCAR initiative on accelerated plant breeding. So they are suggesting how the early testing trial, they can be incorporated, they can advance the rapid recycling of the parents. For that purpose, they have a protocol how we can, Dr. James also mentioned already about this genomic estimated breeding values, how that can be helpful. I don't want to go detail about the GS models and all, but definitely if you can integrate the genomic selection along with RGA, so we are very hopeful that we will be able to, gain genetic. We will be able to achieve the target of doubling the genetic gain for various traits in the coming days. So I'll rest my talk here. Thank you so much for your patience.
Thank you. Any questions for? Yes.
Ashutosh, I must congratulate you for the wonderful presentation. My question is, unlike other RGA, here the duration reduction is specifically focused during flowering to pod filling. Is there a reason why it is going in that stage?
Yeah, because as I mentioned that we have tried a different permutation and combination with different intensity of light as well as photo period as well as temperature in the past. So maybe you are also a part of, we have tried to take some lessons from Pigeon P as well. But in case of groundnut, from still starting from sowing till flowering, the duration we are not able to reduce. Maybe due to the day neutral nature of the crop. So that's the reason why we are, and our intention is to harvest as soon as possible the immature kernels maybe. So the the time frame, that the window that we are left with is the flowering to pod filling. That's why we are targeting that.
All right, Haley. Yes, ma'am.
Thank you for a nice presentation. I think this is critical on the way forward. I just want on the way forward that you indicated, we would like to evaluate under different temperature regime and the range from 25, 30. Probably would it be useful to increase it a bit? And under natural condition, for example, a variety called Samna 24, released in West Africa. When you grow it in Ghana, it takes around 95 to 100 days. But when you take it to Niger, which is a bit hot and a bit dry, it matures 75, 80 days. And they call it actually extra early, while it was released as sort of short duration. That's one aspect. And you said currently, 8 to 10% of your population is going through this RJ facility. Have you considered probably reducing the number of progenies per cross such a way that you could increase the percentage, number of crosses you take through RJ, because you are using elite by elite, crosses. Probably you have a closed population development program where you may not need large number of F2s per cross.
Yeah, thanks for the question. So we are developing the GS pipeline for that matter. So for different traits, we have sequence already, and we are developing GS models. So for that question that you have mentioned, can you reduce the number of progeny of that process that is coming out? So GS prediction that can be quite handy in this aspect, I guess. So in the in the future, I am hopeful that we'll be soon doing that.
Just little addition to Ashutosh on the reducing the time period for maturity. So in groundnut, we studied early, late, and medium maturity. In flowering, there is no difference. Big difference is from flowering to pod maturity. And that to reduce that, lighting will not help. It's a temperature which will help. So some of the station where it is maturing early, studied, the night temperature has some pattern and it has certain threshold. We are going to apply that one and see if we can.
Yeah, as I mentioned already, we have a clue from Dr. Ajay as well. So the Tamil Nadu one place that we have a temperature pattern we are trying to implement it. Thanks for that.
Thank you, Ashutosh. Please give him a round of applause. Let me thank all the 6 presenters of this session coming from China, Japan, Malawi, and India. And also the audience for asking some fantastic questions and starting that conversations. And let me thank my co-chair, Dr. Naveen Puppala, for conducting this session. I just want to make a few remarks in the context of this AAGB, which started with the sequencing of the genomes of and progenitors and also the cultivated groundnut and the way we are today. I mean, the expectation was that these genomic tools to be used and applied in the breeding science so that the cultivars to fit to the farmers, agroecologies and the markets would be developed. And we see that happening in this presentations. Having said that, I'm sure you'll all agree that we are overwhelmed with the amount of genomics data and information we have, and then we are really starving on the phenotype data. So I think it's the time to bring that up here so that we make the meaning of the genome sequences and make it to the real applications of the breeding. And as Ashutosh has concluded, I think RGA combined with genomic selection would be the future for groundnut. And then I think soon we should be moving away from the marker selection for the reason that most of the traits we target in the breeding are quantitative traits. Thank you.
Ladies and gentlemen, let's give a round of applause for all the speakers for their wonderful talk, and the chair and co-chair for conducting this session. Thank you. Ladies and gentlemen, here we close this session on genomics assisted breeding and close this day 2. Please move forward for the coffee and refreshments, and we'll retreat after. Tomorrow we'll be again joining here, and looking forward for your participation for the final day. We are having sessions on quality and followed by frontier technology in groundnut, bringing on breakthrough solutions as well as translating genetics and genetics into groundnut cropping systems. So looking forward for your participation, and we retreat here. Thank you, one and all.